profile, and then we can start the session.
ನನಗೆ co-host ಅಥವಾ ನನಗೆ sharing rights ಕೊಡ್ಲಿಕ್ಕೆ ಹೇಳಿ ಸಿದ್ದೇಗೌಡ್ರಿಗೆ.
ಮಿಸ್ಟರ್ ಆಂಟನಿ ಅವರು ಸಿದ್ದೇಗೌಡ ಅವರೇ, ಇವ್ರಿಗೆ ಸರ್ಗೆ ಪ್ರೆಸೆಂಟರ್ ಇದನ್ನ ಕೊಡಿ.
ಕೊಟ್ರು. ಥ್ಯಾಂಕ್ ಯು. ಮಕ್ಕಳೊಟ್ಟಿಗೆ ಇಂಟ್ರಾಕ್ಷನ್ ಆಗುತ್ತೆ ಹೇಗೆ?
ಮಾಡನ್ ಸರ್, ಇಂಟ್ರಾಕ್ಷನ್ ಡೌಟ್ ಸರ್, ಕ್ವಶನ್ಸ್ ಕೇಳಿದ್ರೆ ಚಾಟ್ ಬಾಕ್ಸ್ ಅಲ್ಲಿ ಹಾಕ್ತಾರೆ.
ಎಷ್ಟು ಜನ ಇದ್ದಾರೆ ಮಕ್ಕಳು? ಜಾಯ್ನ್ ಆಗಿದಾರ
ಸರ್? ಸದ್ಯಕ್ಕೆ ಜಾಯಿನ್ ಆಗ್ತಾ ಇದಾರೆ. ಕ್ಲಾಸ್ ಟೈಮ್ ಇರೋದ್ರಿಂದ ಯೂಟ್ಯೂಬ್ ಅಲ್ಲಿ ನೋಡ್ತಾರೆ ಸರ್. ಆಮೇಲೆ ಕೆಲವೊಂದು ಜನ ಜಾಯಿನ್ ಆಗಿದ್ದಾರೆ ಈಗ.
ಎಷ್ಟು 3 ಜನ ಇದ್ರ ಅಷ್ಟೇನಾ?
3 ಜನ ಇದ್ದಾರೆ ಸರ್ ಸದ್ಯಕ್ಕೆ. ಇದು ಕ್ಲಾಸ್ ಟೈಮಿಂಗ್ಸ್ ಸ್ವಲ್ಪ ಕನ್ಫ್ಯೂಸ್ ಆಗ್ತಾ ಇದೆ ಅಂತ ಹೇಳ್ತಾ ಇದ್ದಾರೆ.
Next is a content creation company that can turn your ideas.
ಶುರು ಮಾಡೋಣ ಸರ್? ಶಾಲ್ ವಿ ಸ್ಟಾರ್ಟ್ ಸರ್?
Yes, please.
ಎಲ್ಲರಿಗೂ ಶುಭ ಸಂಜೆ. ವೆರಿ ಗುಡ್ ಈವ್ನಿಂಗ್ ಟು ಒನ್ ಅಂಡ್ ಆಲ್. ಇವತ್ತಿನ ಪ್ರತಿಭಾಶೋಧ ಉಪನ್ಯಾಸ ಕಾರ್ಯಕ್ರಮಕ್ಕೆ ನಮ್ಮೊಂದಿಗೆ ಡಾಕ್ಟರ್ ಬೆಲ್ಲೆ ದಾಮೋದರ್ ಶೆಣೈ ಸರ್ ಇದ್ದಾರೆ. ಅವರು ಫಂಗಲ್ ಟ್ಯಾಕ್ಸಾನಮಿಸ್ಟ್ ಆಗಿ ಕಾರ್ಯನಿರ್ವಹಿಸ್ತಾ ಇದ್ದಾರೆ. ಇವತ್ತಿನ ಉಪನ್ಯಾಸ ಶುರು ಮಾಡೋದಕ್ಕಿಂತ ಮುಂಚೆ ಅವರ ಬಗ್ಗೆ ಒಂದು ಕಿರುಪರಿಚಯ ಮಾಡಿಕೊಡ್ತೀನಿ. Dr. Belle Damodara Shenoy is a fungal taxonomist by training. After completing his bachelor's and master's degree from India, he pursued his PhD in fungal taxonomy in the University of Hong Kong under the guidance of Professor Kevin Hyde and Dr. Rajesh Chivan. His PhD thesis titled Multigene Phylogeny of Selected Anamorphic Ascomycetes dealt with the evolutionary relationships of sexual genera such as Porydesmium complex and provided insights into the implications of their polyphyly in the fungal taxonomy. Dr. Shenoy, for a brief period of time, worked as a postdoctoral fellow in Dr. Keith Stafford at the Agriculture and Agri-Food Canada, Ottawa, on DNA barcoding Fusarium in 2008. Dr. Shenoy returned to India and joined the Microbial Type Cultural Collection, MTCC, CSIR Institute of Microbial Technology, which is located at Chandigarh. It is well known as CSIR and INTEC. He supervised 2 PhD students at CSIR INTEC and published several high-impact papers on fungal taxonomy, phylogeny, DNA barcoding, and comparative genomics. In 2013, Dr. Shenoy moved to CSIR National Institute of Oceanography, Goa, and initiated research on diversity and taxonomy of microbes, which is associated with the carbons known as crude oil derivatives. He is presently working at CSIR NIO Regional Center, Visakhapatnam, Andhra Pradesh, and supervising 2 more PhD students on bacterial diversity in select marine niches. Dr. Shenoy has so far published 45 papers in reputed international and national journals. He is the founding member and presently the managing editor of a new mycology journal, Myco-Asia Journal of Modern Mycology. You can visit the website at mycoasia.org. Additionally, Dr. Shenoy has been a member of the International Subcommission on Calectotricum taxonomy. Dr. Shenoy is on the advisory board of MIT Institute of Oceanography and Atmospheric Sciences, India. His non-scientific hobbies include traveling and exploring India and writing poems. With this brief introduction, I once again welcome Dr. Belle Damodara Shenoy sir. Also, I would like to welcome formally all the teaching faculty and student fraternity who have joined with this webinar series in this WebEx platform as well as YouTube channel. With this brief introduction, once again, I welcome Damodar sir. Over to you, sir.
Thank you, Umesh avare. Thank you very much for the kind introduction, and thank you, KST, for inviting me to give this talk on mycology. I am a mycologist, as introduced by Mr. Umesh. I would like to spend next 30 to 45 minutes talking about mycology, the study of fungi. I hope my voice is clear and loud. If it is not clear and loud, please let me know. I
Yes,
can
sir.
fix.
Now it is audible clear, sir. Thank you.
Yeah, okay. For the sake of avoiding distraction, let me switch off the video and let me start presenting with my slides. Can you see my slides, sir?
Yes, sir. Yes, sir. You can see an introduction to mycology. Please go ahead, sir.
Okay, thank you so much. Yeah. Um, the I'm going to spend some next 30 to 40 minutes talking about the awareness about fungi, basic information that even a common person, a common citizen should know about fungi. Then I will move on to the basics of fungal biology, some technical issues, technical terms, technical definitions. And at the end, I will talk about the applications of fungi in our day-to-day life. See, let me start with the name. How do you call fungi in local languages? For example, in Kannada, how do we call fungi? Yeah.
Sir, sir, you both speak in Kannada and English, sir.
ನಾನು ಕನ್ನಡದಲ್ಲೂ ಮಾತಾಡ್ಬೇಕೀಗ.
ಮಾಡಿ ಸರ್, ತೊಂದರೆ ಇಲ್ಲ ಸರ್, ಮಾಡಿ ಸರ್ ಪ್ಲೀಸ್.
ಪಿ ಯು ಸಿ ಮಕ್ಕಳು ಇದ್ದಾರ ನೋಡಿ. ನಾನು ಕನ್ನಡ ಮತ್ತು ಇಂಗ್ಲಿಷ್ ಎರಡರಲ್ಲೂ ಟ್ರೈ ಮಾಡ್ತೀನಿ. ನಾನು ಕನ್ನಡ ಅಷ್ಟೇ ಚೆನ್ನಾಗಿಲ್ಲ, ಎಸ್ಪೆಷಲಿ.
ಇರ್ಲಿ ಸರ್, ಪ್ಲೀಸ್ ಸರ್, ನಿಮ್ಗೆ ಎಷ್ಟು ಇದಾಗುತ್ತೆ ಅಷ್ಟು ಮಾಡಿ
ಸರಿ ಸರಿ ಸರಿ. ಹಾಗಾದ್ರೆ ಈ ಒಂದು ಪ್ರೆಸೆಂಟೇಶನ್ ಅಲ್ಲಿ ಮೊದಲನೇದಾಗಿ ನಾನು ನಮ್ಮ ಫಂಜೈಗಳ ಬಗ್ಗೆ ಸಾಮಾನ್ಯ ಜನರಿಗೆ ಏನು ಗೊತ್ತಿರಬೇಕು ಅದರ ಬಗ್ಗೆ ಸ್ವಲ್ಪ ತಿಳುವಳಿಕೆ ಕೊಡ್ಲಿಕ್ಕೆ ಪ್ರಯತ್ನ ಮಾಡ್ತೀನಿ. ಎರಡನೇದಾಗಿ ಫಂಗಲ್ ಬಯಾಲಜಿ ಅಥವಾ ಮೈಕಾಲಜಿ ಬಗ್ಗೆ ಕೆಲವೊಂದು ಬೇಸಿಕ್ ಇನ್ಫಾರ್ಮೇಶನ್ ನಿಮಗೆ ಕೊಡ್ತೀನಿ. ಕೆಲವೊಂದು ನಿಮ್ಮ ಪಠ್ಯ ಪುಸ್ತಕಗಳಲ್ಲಿ ಇರ್ಬೋದು, ಆದ್ರೂ ಅವುಗಳ ಬಗ್ಗೆಯೂ ಹೇಳ್ತೀನಿ ಮತ್ತು ಪಠ್ಯ ಪುಸ್ತಕದಲ್ಲಿ ಇರುವಂತಹ ವಿಷಯಗಳನ್ನು ನಿಮ್ಗೆ ಹೇಳಲಿಕ್ಕೆ ನಾನು ಪ್ರಯತ್ನ ಪಡ್ತೀನಿ. ಕೊನೆಯದಾಗಿ ಫಂಜೈಗಳ ಅಪ್ಲಿಕೇಶನ್ಸ್ ನಮ್ಮ ಸಾಮಾನ್ಯ ಜೀವನದಲ್ಲಿ ಹೇಗೆ ಇರುತ್ತೆ ಎಂಬುದರ ಬಗ್ಗೆ ಹೇಳ್ತೇನೆ ನಾನು. ಮೊದಲನೇದಾಗಿ ಕನ್ನಡದಲ್ಲಿ ಫಂಜೈ ಅಂದ್ರೆ ಏನು ಅಂತ ನಿಮ್ಗೆ ಹೇಳಲಿಕ್ಕೆ ಪ್ರಯತ್ನ ಪಡ್ತೀನಿ. ಹಾಗೂ ಹಿಂದಿಯಲ್ಲಿ ಫಂಜೈಯನ್ನು ಏನು ಕರೀತಾರೆ ಅಂತ ಹೇಳ್ತಾರೆ. ಕನ್ನಡದಲ್ಲಿ ಫಂಜೈ ಅಂದ್ರೆ ಶಿಲೀಂಧ್ರ. ಫಂಜೈ ಆರ್ ನೋನ್ ಆಸ್ ಆರ್ ಕಾಲ್ಡ್ ಆಸ್ ಶಿಲೀಂಧ್ರ ಇನ್ ಕನ್ನಡ ಲ್ಯಾಂಗ್ವೇಜ್. ಹಾಗೆ ಒಂದು ತರಹದ ಫಂಜೈ ಇರುತ್ತವೆ, ಅಣಬೆಗಳು. So these are like, let me just get going with English, then I will come back to Kannada after some time. So in Kannada, fungi are known as Shilendra, okay, but some reproductive structures known as mushrooms are known as anabe in Kannada, okay. Similarly, in Hindi, fungi are known as kavaka, okay. And the mushrooms are known as kukurmutta, madhurika, so like that. If you didn't know, singular form of singular form is fungus. The plural form is fungi. Okay, and there could be three pronunciation for this term, fungi. This can be, first pronunciation can be fungi, second can be fungi, third could be fungi. Okay. As you may know, fungi or fungi can be poisonous, but there are also many beneficial aspects of fungi that I would like to highlight in this presentation. ಉಮೇಶ್ ಅವರೇ ನಾನು ಮಧ್ಯದಲ್ಲಿ ಕನ್ನಡದಲ್ಲಿ ಹೇಳ್ತೀನಿ ಬಿಕಾಸ್ ಮೈ ಫ್ಲೋ ಇನ್ ಇಂಗ್ಲಿಷ್
Whatever is convenient for you, sir. Please go ahead.
yeah, because my flow in Kannada is not very good. I tried for a minute and I felt that I'm not up to the mark. Okay,
No, it's okay, sir.
please excuse me for that.
Please go ahead.
Yeah. But in between, I will try to explain in Kannada. Okay, whenever possible. Okay.
Noted, sir. Please.
Please. Thank you. Um, uh, for practical purposes, fungi can be divided into three groups. First one is mushrooms. Second one molds, third one yeasts. Okay, mushrooms are these beautiful structures, reproductive structures, macroscopic structures, ಕಣ್ಣಿಗೆ ಕಾಣುವಂತಹ structures. These are macroscopic and they are multicellular. When it comes to molds are the kind of microscopic fungi present in dust particles on bread and all. These are microscopic and they are multicellular. For example, aspergillus is a mold. Actually, when it comes to mushroom, there are many mushrooms like button mushrooms are there, ganoderma is there. And third form is yeast. These are mostly unicellular. When they divide, they become bicellular. For example, baker's yeast, Saccharomyces cerevisiae, and all those stuff. During the COVID second wave, can you see my slide? Why I'm not able to see my slides here?
No, sir, we can see. I think it is minimized. You can maximize that.
Okay, now I can see. Thank you.
We can
Thank
see.
you. Thank you. Sorry about that. During the second wave of COVID, you might have definitely heard about black fungus. And because this black fungus expression is very wrong, because the fungus here is highlight, but the symptoms caused by that fungus are black. Therefore, the fungus was incorrectly known as black fungus actually. That shows the kind of ignorance we have about mycology in our day-to-day life. Unfortunately, this so-called black fungus was responsible death of around 4,000, 5,000 people in India who were immunocompromised or who had COVID infection. Not only during COVID time, even fungi play important role in our day-to-day life. For example, skin infections. Fungi cause can cause severe fungal severe skin infection. For example, this ನಂಜು ಅಂತ ಹೇಳ್ತಾರಲ್ಲ, or athlete foot, especially in monsoon seasons, you might get athlete foot. Yeah. And even on common bread, actually, we see this kind of fungi. These are known as bread mold. So I am showing you a small video clip. Just so have a look at it. It is just it's a video clip of bread mold in action. A video of just 10-15 seconds. Just have a look at it. You can see the progression of the growth of fungi. Let's put growth of fungus on growth of fungi on bread actually. Here you can see citrus mold in action. It's a fungus, which is a kind of spoiling the orange. Okay, of course, fungi cause food spoilage, but some groups of fungi are also edible. For example, mushrooms. Mushrooms are edible. I will talk about more, or I will talk more about it later stage of this presentation. See, even fungi play important role in milk products. For example, the taste, and the fungi are involved in making of cheese. These are very simple things just to get your attention, towards this presentation and towards mycology. So we can also find different kinds of fungi in forest. I hope you can see the video. Can you hear the voice?
No, sir.
Yeah,
Video
no?
is not.
ವಾಯ್ಸ್ ಕೇಳಿಸ್ತಾ ಇದೆಯಾ ಇಲ್ವಾ?
No, sir. Video voice is not coming.
ಏನ್ ಮಾಡೋಣ ಇರ್ಲಿಯಾ? ಜಸ್ಟ್ ಪ್ಲೇ ಮಾಡ್ತೀನಿ ನಾನು. ಮತ್ತೆ ನೀವು
Yes,
ಮತ್ತೆ
sir.
ಯೂಟ್ಯೂಬ್ ಅಲ್ಲಿ ನೋಡಿ, ಸರಿನಾ? ಓಕೆ.
Yes, sir. Please share that link in the chat box also, sir, later.
Later I will share. Yeah. I mean, that was a spectacular video on mushrooms, especially macrofungi from the forests of northeast India, and those were captured by two Australian movie makers or photographers, Stephen Axford and Catherine Machiniak. I hope you enjoyed that. You can go back to this video on YouTube and just you can watch it with the beautiful music, background music also. Excuse me. Okay, moving on, let me just take you to the little bit difficult part of mycology, basics of fungal biology. I'll try to explain it in a very simple words. See, if you look at the tree of, if you see the diversity of life on this earth, all that diversity can be represented in the form of a tree. And like any tree, there would be a root. In this case, there would be an hypothetical common, hypothetical oldest ancestor here. And here you have the kingdom of fungi represented by a mushroom, and you have animals here, and you have plants here. So you study fungi in botany. Actually, fungi are closer to animals than actually to the plants. This is based on the genetic data. And that is based on genetic data. I mean, basically not genetic data, this is multiple protein data actually. It's on molecular data. If you did not know, the largest living organism on this earth is a fungus. You know, it's not a whale or anything, it is a fungus. Let me give you more information about it. It is a Guinness World Record. And it's a Guinness World Record because this particular fungal mycelial network covers a total area of 965 hectares, which is equivalent to 1,350 soccer fields or football stadium actually. So how a fungus can be so big, so vast? Basically, if you say the name of the fungus is honey mushroom, its scientific name is Armillaria austraea. This is reported from United States. Basically, even though mushroom is a macroscopic structure visible to our eye, but the below ground is a mycelial network. In this case, the mycelial network is covering a total area of 2,385 acres. I mean 2,385 acres. As I told you, it is as big as 1,300 a football stadium. And the mycelial network is around 1 meter below the ground, underground. At least that means 3 feet underground. So it is calculated that this particular fungus or mushroom could be at least 2,400 years old. That means this fungus has been living for last 2,400 years. I mean, and this is as per the Guinness World Record. This is the largest living organism at the moment. About the largest living organism as a fungus is a fungus. Another interesting fact about fungi is that it's believed that life originated in an aquatic system, in a marine system. Then the preliminary life came onto the land. Before even plants could came onto the land, fungi were already there. They had already inhabited the land even before the plants did actually. So they not only had inhabited the land even before the plants did, they dominate in the ancient world. There are some evidences which suggest that uh, there were mushrooms as tall as 8 meters, during that period. And but afterwards, there was a competition between giant mushrooms and the plants, and giant mushrooms lost out in the battle and started dominating. But just remember that the evidences suggest that the fungi had inhabited the land even before the plants did. Okay. Coming to the definition of fungi, fungi are eukaryotic. That means their nuclear envelope is very, very definite. They have no plastids, photosynthetic pigments. They are heterotrophic. That means they are not able to make their own food because they don't have they don't have photosynthetic pigments. They are osmotrophic. They absorb rather than ingest food. They are opportunistic. And when they are opportunistic, they are capable of exploiting many different substrates. They can exploit substrates like plastic, crude oil. Fungi are never amoeboid at any stage, and I'll I'll give more details about their minute structures, mycelia and stuff. Uh, Fungi reproduce by unicellular or multicellular spores. They generally have haploid somatic nuclei, and most fungi mostly do not produce flagellate cells. And the it's a branch of science which studies fungi is mycology. So this is the basic structure of the fungus. If, for example, if this is a fungal spore, when it starts germinating, there will be a germ tube. The germ tube starts elongating and these become a hyphae, and the many hyphae come together to form a mycelium. So spore, hypha, mycelia, these are the three important terms when it comes to. So this is the just a graphical representation of a fungal spores. Here you can see hypha, and the spores are attached to them. And this is one more animation of how the fungal spores are released. It's just an animation. Now coming to the classification of fungi, as I told you, fungi were studied in the botany, and they are still being studied in botany. But once the fungi were subkingdom, were a subkingdom under plant kingdom actually. Okay, and that time subkingdom fungi included both plasmodial and non-plasmodial form of fungi. But in 2007, the taxonomists revised the taxonomy of fungi. They revised based on DNA sequence rate and morphology. Now the fungi are redefined phylogenetically based on the evolutionarily important characters. So you don't have traditional Chytridiomycota. These were found to be not a natural group of or a natural group of or organisms. So Chytridiomycota is used in a very restricted sense, and there is no more Zygomycota. Traditional Zygomycota was found to be polyphyletic. There is no more traditional Zygomycota. Deuteromacids were taught as Deuteromacids were never taught as a valid taxon. They were just a group of fungi which produce only asexual structure. I'm not sure this is taught in you or taught. This is taught in your 10+2 or 10+1 classes, but it is good to know the updated information about fungal classification. For more information about fungal classification, you can refer to this paper by Hibbett et al. Published in 2007. Another important question is one of the important questions is how many fungal species are there? I mean, we cannot definitely tell how many are there, but we could make a guess. I mean, if I were to make a guess, I would say maybe 15 lakhs to 45 lakhs. That would be my guess. But even other mycologists have similar thoughts. Hawksworth estimated that there could be at least 15 lakhs fungal species on this earth, and we have so far estimated only not estimated. We have so far described only 2 lakhs fungal species. And where are the missing fungi? Where can we find the missing fungi? We could find the missing fungi probably in poorly studied studied areas or substrates or the regions of India or tropical countries, or maybe in the oceans. When it comes to mushrooms, it is believed that there could be at least 27,000 species of mushroom species, and we have so far described only 14,000 species of them. So out of 14,000 species of mushrooms, only 2,000 species of them are edible. So for remaining 12,000 species of mushrooms, we don't have enough information or edibility actually. So either they are we would just stay. It is better to stay away from such mushroom. This is a paper which describes the list makes a list of 2,100 edible mushroom species from the world actually, and this list is not perfect, but this gives an idea about the number of edible mushroom species in this world. And mushroom market is a big market. Global mushroom market is valued at around 46 billion US dollars. So that is the current market size of mushroom mushroom market at the global level, 46 billion US dollars. And this market is mushroom market is gonna grow at least 10% per annum, actually, at the rate of 10%. There is a great scope in mushroom cultivation, not mushroom production, cultivation, mushroom selling, mushroom marketing, and value-added products. So this website gives 18 popular types of edible mushrooms. Let me just give the names of all of them. This is the first one is cremini, morel, shiitake, oyster mushroom, king oyster mushrooms, lion's mane, enoki mushrooms, white button mushrooms, portobello mushrooms, porcini mushrooms, maitake mushrooms, and matsusake mushrooms. Reishi mushrooms, giant puffball mushrooms, bunashimigi mushrooms, use ear, straw mushroom, chanterelle. These are the 18 popular commercially available edible mushrooms. Of course, fungi are known for their destructive plant diseases, but fungi also collaborate with other life forms. For example, fungi collaborate with the plant root system, then they are called mycorrhizal. So mycorrhizal partnership is more about nutrient exchanges and the communication between mycorrhizal fungus and plants. So when it comes to lichens, where you have a fungal partner and you have a algal partner or a cyanobacterial partner. So lichens are a good bioindicator of climate change. You may know that fungi are a great source of medicines. You might be aware that the first antibiotic, penicillin, is from a fungal origin, and Alexander Fleming received a Nobel Prize for the discovery of penicillin, along with two other researchers. The current name of the penicillin producing strain is Penicillium rubens. When I was a student, I was taught that Penicillium notatum was the name of the fungus which produced penicillin. Now the name has changed. Now the name is Penicillium chrysogenum. So we taxonomists have interesting way of doing our taxonomic work. Now we use DNA sequence data. Based on the DNA sequence data and morphology, we have identified or renamed that particular fungus as Penicillium rubens. Okay. And penicillin is not the only medicine that we get from fungi. This is a list of FDA approved, FDA USA approved medicines from fungi. This include many applications like cholesterol lowering, for combating bacterial infection, to treat terminal illness, to be used during organ transplantation, or to treat fungal infection, or for that matter to treat migraine headaches. All these kinds of medicines are from fungi. Actually, the compounds are listed here. Okay. So that means the fungi are a great source of medicines for mankind, actually. So if somebody is interested in studying fungi and the molecules from the fungi, so it would be an interesting area of research for youngsters. So in marine environment, you can find fungi in many places like driftwood, marine woods, roots, nematophodes, seedlings, leaves of mangrove plants, soils, sediments in marine environments, seawater, and dead and decomposing animal substrata. Okay. We have been working on a marine pollutant called tarbo. Let me show you a small video about it. Okay, you might have seen in that video, there are some black kind ball-like structures. So what are those black things, I mean, those are tar balls. Those are kind of a marine pollutants. Really. And the second question you might ask, who put those black structures or tar balls on the beach? Nobody put them there. They have come from the open ocean. Uh, I'll explain more about it later. And the question is, don't they spoil the beach? Of course, the star balls spoil the beach. The important question is, are they safe to humans? They may not be safe to humans if when they come in touch with when humans come in touch with them. Actually, it might cause allergic reactions because of exposure to some potentially pathogenic microbes, including pathogenic fungi. Tar balls, these are tar balls. These are semi-solid blobs. These are solid outside, liquid inside, soft and sticky, brittle or hard. They are black to brown in color. They are millimeter to centimeter in size. They are seasonal appearance in India. Basically, they are found on the west coast of India. In Karnataka, you can find these. Not find. They come. These are reported from the beaches of Kundapur, Mangalore, Karwar. Also, in other states like Goa, Maharashtra also. So it's not just India's problem. It's a tarballs are a global problem. These kind of tarballs have been reported from all over the world, which is from the many parts of the world. These tarballs contain many microbes, including fungi, bacteria, plankton, viruses. We studied bacteria and fungi. In this presentation, I will talk about only fungi. Okay, and we wanted to know what kind of fungi are associated with tarballs. So these are the kinds of fungi we isolated from tar balls, and we did the ITS sequencing when we extracted DNA from this the fungal material, fungal mycelia, and we amplified a gene region called ITS, and we did the phylogenetic tree construction. This is a phylogenetic tree, and we actually placed these fungi on the tree of life. Actually, we identified them based on the DNA sequence data. This is the first time in the world somebody used a DNA ITS sequence data to characterize the fungi associated with tarball. We continued our work, and we found more number of different kinds of fungi associated with tarballs. This is more about research. I don't want to complicate you with the details. But last but not the least, if you are interested in mycology, you could start browsing social media for interesting stuff on mycology. First, you could go to Planet of Fungi Facebook page, where Stephen Axford and Catherine Marcinek post interesting pictures about mushrooms and also other macrofungi. I hope some of you might have seen the movie Fantastic Fungi. So they have a Facebook page. There is something called Fungi Film Festival. It happens every year. So you can go to their Facebook page and just get more information about movies on fungi. With this, I conclude my presentation. I thank KSTA for giving me this opportunity, and also thank my employer CSI RNIO, and I thank my students and collaborators, and thank today's audience for your patient hearing. These are my contact details. You can always email me if you have any queries about mycology. I will be happy to answer. Thank you so much.
Thank you very much, sir. If there are any questions, I request students to post in the chat box or mention in the Q&A box, or they can raise their virtual hand so that we can unmute them. They can directly speak to sir.
Yeah, I would be happy to answer. I would be happy to interact. About the YouTube video, I will share the link. Sure.
Sir, meanwhile, there is a question.
Yeah, just give me a second. Let me
Yes,
share the link actually, please.
Yes, sir, please.
Okay. I shared the link to that YouTube video. Yeah. Now, what is the question?
So how can we morphologically differentiate poisonous and non-poisonous fungi?
Quite difficult. Even for that matter, even for that matter, even if you use molecular data, you cannot tell between a poisonous mushroom from a non-poisonous one. And there are no standard scientific methods to differentiate an edible mushroom from a non-edible or a poisonous one. Basically, it's a traditional knowledge. Edibility is a traditional knowledge. We inherit from our ancestors. They might have tried, they might have done trial and error. They found out this particular variety is edible. Members of the same species can sometimes differ in their edibility actually. So there is no standard. That makes a little bit matter complicated. People assume that people misidentify mushrooms and and they get poisoned actually. Especially there are many reports of mushroom poisoning from Assam state of India. A very good question. So maybe you could go to this paper from 2021, where they have listed 2,100 edible, mushrooms. I hope I have answered the question.
Sir, yes, sir. Thank you very much, sir. Sir, any biomass production of fungi is taking place in India for the research purpose and other industry purpose usage?
I don't understand your question. I mean, biomass production, I mean, it depends upon your purpose of research purpose, right? If you are, you can do large scale fermentation and grow fungal biomass for extraction of, extraction of biomolecules. So that is a very routine process now.
Okay, sir. So it has become a routine for fungi biomass production in India. Okay.
Yes, yes.
So any other questions from the audience? I think questions are over, sir, as of now.
Okay, thank you so much. Yeah.
Thank you very much, sir. On behalf of KSTA and on behalf of all the students and teaching faculty, I would like to thank you immensely for giving a talk on fungi and mycology, sir.
Thank you so much. Yeah.
Thank you so much. Now I request Sri Siddhagowda to introduce the next speaker, Netrvathi madam, to deliver the talk on evolution.
Netrvathi madam ಅವರ
Yes, sir.
ಶೇರ್ ಮಾಡ್ಕೊಳ್ಳಿ ಮೇಡಮ್ PPT.
Sorry, sir. Sir, audible?
Audible and visible, no problem. Nice.
ಕಾಣಿಸ್ತಿದ್ಯಾ?
ಕಾಣಿಸ್ತಿದೆ ಮೇಡಮ್, ಫುಲ್ ಸ್ಕ್ರೀನ್ ಮಾಡ್ಕೊಳ್ಳಿ.
ಹಾ. ಸರ್ ಕಾಣಿಸ್ತಿದ್ಯಾ?
Yes, madam.
Very good evening, madam.
Good evening, sir.
Thank you for accepting our invitation to deliver this evolution talk for PU students.
Yes, sir. Yes, sir. I'll try my best to cover it in 1 hour, sir.
Once again, I welcome you.
Yeah, thank you.
For CST and on behalf of all the attendees.
Yes, sir. Yes, sir.
For starting the session, I request Sri Siddegowda to introduce you, please. Over to you, Sri Siddegowda.
Good evening, ma'am.
Good evening, sir.
ಸೊ ಎಲ್ಲರಿಗೂ ನಮಸ್ಕಾರಗಳು. ಸೊ ಎರಡನೇ ಸೆಷನ್ ಅಲ್ಲಿ ನಮ್ಮ ಶ್ರೀಮತಿ ನೇತ್ರಾವತಿ ಮೇಡಮ್ ರವರು ಎವಲ್ಯೂಷನ್ ಎಂಬ ವಿಷಯದ ಬಗ್ಗೆ ಉಪನ್ಯಾಸ ನೀಡಲಿದ್ದು, ಸೊ ನೇತ್ರಾವತಿ ಮೇಡಮ್ ರವರನ್ನ ಈ ಒಂದು ಉಪನ್ಯಾಸ ಕಾರ್ಯಕ್ರಮಕ್ಕೆ ಸ್ವಾಗತಿಸುತ್ತಿದ್ದೇನೆ. ವೆಲ್ಕಮ್ ಟು ಮ್ಯಾಮ್.
Welcome. Thank you, sir. Thank you, sir.
ಸೊ ಅದೇ ರೀತಿ ನಮ್ಮ ಅಕಾಡೆಮಿ ಮುಖ್ಯ ಕಾರ್ಯನಿರ್ವಹಣಾಧಿಕಾರಿಗಳಾದಂಥ ಡಾಕ್ಟರ್ ರಮೇಶ್ ಸರ್ನು ಕೂಡ ಸ್ವಾಗತಿಸುತ್ತಾ, ಹಾಗೂ ನಮ್ಮ ಅಕಾಡೆಮಿಯ ಅಧ್ಯಕ್ಷರಾದಂಥ ಪ್ರೊಫೆಸರ್ ಅಯ್ಯಪ್ಪನ್ ಸರ್ ಅವರನ್ನು ಕೂಡ ಸ್ವಾಗತಿಸುತ್ತಾ, ಈ ಒಂದು ಉಪನ್ಯಾಸ ಕಾರ್ಯಕ್ರಮಕ್ಕೆ ಕಾಲಾವಕಾಶ ಕಡಿಮೆ ಇರುವ ಕಾರಣ ಸೊ ನೇರವಾಗಿ ನಾವು ನೇತ್ರಾವತಿ ಅವರನ್ನ ಈ ಒಂದು ಕಾರ್ಯಕ್ರಮದಲ್ಲಿ ಓವರ್ ಟು ಯು ಮ್ಯಾಮ್.
Thank you, sir. Start ಮಾಡದ sir?
Yes, ma'am. Over to you.
Okay. Good afternoon, one and all. This is Netrvathi from Government Girls College, Alur, who's going to be delivering a talk about evolution. So evolution, in this 1 hour 15 minutes, we are going to be covering topics related to origin of life, like how life originated on Earth, and evidences for evolution. What are the evidences to say that evolution has occurred on Earth? And we have some theories, very, very basic theories, to explain that, explain how evolution would have occurred. And lastly, I would like to tell the story of human, that is evolution of man. First of all, when you see this cover page, you can see that I have put on three pictures. One is a picture of primordial Earth. So the first picture, what you can see in the slide, is primordial Earth, where we can see there is no life at all. We can see the meteorites are falling on the Earth. We can see volcanic eruptions which are occurring on all the side of the Earth. We can see very little amount of water, very, very little amount of water, and the Earth was unsuitable for life. Then we also have a picture which I'm going to be discussing in the next slides. And this is the story of human evolution. He started with 4 and ended up with 2 legs of walk. So let us move on to the next slide. How actually life originated on Earth. As I said in the beginning, there was no life at all. There was no microorganisms, nothing, because the life on Earth was impossible, because it was a very high temperature. The temperature were very high, and lots of volcanic eruptions, and everywhere we could see the poisonous gases, that is hydrogen, cyanide, and other gases. Now, how actually, before going to the existence of Earth, how actually the whole universe existed. So if you talk about how the universe existed, so there is a theory what is being proposed called as Big Bang theory. So they say that around 15 billion years ago, a structure called nebula. So that nebula had a very big explosion, and because of that explosion, a lot of things were formed in the universe. Like there were stars, there were galaxies, there were planets. So like that, once one fireball called as nebula, it exploded, and that is how we got this universe, a very, very, very big universe, which is having infinite distance. We cannot measure the distance of the universe. So if you talk, that is the theory what is being said. And because of this Big Bang theory, we have our solar system, we have our Milky Galaxy, which has come out about 10 billion years ago. So after the origin of Earth, the origin of life started taking place. So first we got the universe, and inside the universe we got the solar system. Inside the solar system we got so many planets, and then we got, this is the Earth. So first it was the universe, and then the solar system, and then the earth, and then we saw the life on earth. So let us understand how actually life existed on earth. So there are a lot of stories being told to talk about life on earth. So there are many stories, like these are the important theories which are given in the CBSE book or NCRT book. So these are very, very important for the exam. The first one is theory of special creation. So as the name says, special creation means this theory is proposed by the Bible. So in the Bible, they have illustrated that that the life existed because of God. So God created the life on earth, and he took 6 days to create the life on earth. So first day was humans, second day was mother microbes, third day was fungi, fourth day was plants, then fifth day other animals, and seventh, so like that. So he took 6 days for having the life on earth. But this theory is having it is not having any scientific proof to say that life existed in 6 days. So therefore this theory is not so accepted everywhere, but it is discussed in the textbook. But you have the next one is theory of abiogenesis. So abiogenesis means it is the origin of life from non-living things. So this was said by Thales, Aristotle, and Democritus. So what they saw is, they saw that when you keep something fruits, when you keep fruits for longer time, you can see that from the fruit you can see something coming out of that. Or when you keep a shirt for unwashed for a long time, then they saw that there were small mites coming out, small worms coming out from the unwashed cloths. So they said that might be because of non-living things the life existed. But again, this theory is also not accepted because there is no evidence to say that from non-living things the life has existed. So this is a very important theory of abiogenesis, is a very important question which will be asked for 1 mark in your second PUC exam. The next one is theory of biogenesis. So to some extent, yes, this was accepted. It says that the life originated from pre-existing life. Means what? The pre-existing life, whatever the life was there before, that gave rise to the new life on the earth. So this was proposed by Francisco Reddy, and he conducted a lot of experiments where he placed some nutrients in flasks, and one flask was uncovered without any covering. Andre, he took a bowl, he uncovered the bowl and he placed some food in that. Then he took another bowl, he covered that bowl and he placed the food and he covered that bowl. And then he got to know that he kept it for a long time. Then he could see that the uncovered bowl saw some other organisms than the rotten food, but the covered bowl did not show any life. So that means what? The life would have existed from the pre-existing life. But again, Francisco Reddy was not possible to say how the pre-existing life existed. So there they failed. The next, another important theory is cosmic theory or panspermia. This is also very important for the exam. They say, according to this theory, they say that the organisms which are traveling, the materials which are traveling in the space, so meteoroids or cosmids, whatever are traveling in the space, those would have fallen on the earth and they have converted into spores, and those spores have been converted into organisms, and that is how the life existed on earth. But again, there is a lack of evidence if spores had fallen on earth, so this theory is also not accepted. The very most accepted theory till now is theory of chemical evolution, which was proposed by Oparin and Haldane. So Oparin and Haldane proposed that the life existed from the evolution of chemicals. Now, before going to this theory in detail, let me say what is evolution. So evolution means it is nothing but unfolding unfolding or something, unrolling of something. Means what? Some organisms on the earth will change according to the atmosphere, will change according to the environment, will change according to the natural resources what they get, and form into a new species or into a new organism which are totally different from the ancestral organism. So that is what is called as evolution. So what Oparin says that, Oparin says that the chemicals have got evolved. Okay, so that means what? Basically, the primordial earth had hydrogen, oxygen, carbon, nitrogen, and all these. So these chemicals have got bonding with each other, and that is how the life existed on earth. So let us understand how they have come up with. So there are 5 points about chemical evolution. The first one is the primitive atmosphere of the earth. So primitive atmosphere of the earth means here what they say here is the earth was very primitive. So as I showed you the photo here, so you can see here there is a photo which shows how the primordial earth was there. You can see the sun here with lot of explosions in the sun. You can see a dusty atmosphere. You can see a meteorite or a star falling down. You can see earth, and you can see little amount of water. This is called as a dilute soup, where actually the formation of life was taking place. So what according to Oparin is, he says that life existed with 5 steps. First, there was a primitive atmosphere of the earth. The primitive atmosphere was very, very high. The temperature is unimaginable. You cannot imagine the temperature at all. And there was no reduced oxygen. There was no water. There were no clouds. There was no rainfall. Then what happened? Slowly the temperature of earth calmed down. It started cooling down. Why? Why? Because the earth was what? Was not very near to the sun or not very away from the sun. So this made the life to exist on earth. So the temperature of the earth, which was might be about 800 degrees Celsius, started to come down to 500, 400 degrees Celsius. So when the temperature came down to 400, 500 degrees Celsius, then the next step took place. What is that? Formation of micromolecules. The micromolecules were formed. So I have the next slide to show. This was the primitive components which were there in the atmosphere. So there was water vapor, ammonia, hydrogen cyanide. This is a very, very poisonous substance which was found on the primordial earth or primitive earth. Then we had lots of carbon dioxide, hydrogen, and methane. So what happened? These molecules, with basic molecules, carbon, hydrogen, oxygen, nitrogen, sulfur. So these molecules, these started binding with each other and they started forming into micromolecules. These micromolecules further went bonding to form macromolecules. That means, why did this happen? Bonding, how did this bonding happen? Because of the cooling of the Earth's temperature. The Earth temperature started cooling down, which made these micromolecules to form into macromolecules. The macromolecules started binding with each other. So already you know about biomolecules. So biomolecules means the carbohydrates, proteins, lipids, and fats. So these are the vitamins. These are the main macromolecules. So those macromolecules, they started colliding with each other. So that means what? You had the nucleic acid in the middle, which was bound by the protein around it, and that formed into the first part of the life, that is protocells, or you can call it as protobionts. So what are protobionts means? They are nothing but the colloidal system of different macronutrients which were able to live in the environment, which were able to adjust in the environment. So that is the first life what you can see on the earth, and they are called as protobionts or protocells or ebionts. Later on, you can see the origin of life, that is the origin of cells. Later on, we got the prokaryotes. From the prokaryotes, we got the eukaryotes. From eukaryotes, we got heterotrophs, and then came the autotrophs. So let me show you the next slide. So this is the slide which shows how the chemicals have evolved during the course of the evolution. So you can see that this is the sun, and you can see this is the atmosphere of the earth, where you cannot find any clouds at all. There are no clouds. These are nothing but they are nothing but they are not clouds actually, because there was no condensation of water on the earth because of high temperature. 500 degrees, 400 degrees was the temperature, so we cannot expect cloud formation there. So what you can see here is, you can see pyramids are getting converted into nucleoproteins, purines into nucleotides, amino acids are getting converted into proteins, the fatty acids and glycerols are converted into fats, sugars are converted into polysaccharides. So this is what actually happened during the chemical evolution according to Oparin and Haldane. So let us see the next thing. So this gives an overall view of the chemical evolution. So this is the Big Bang theory, origin of universe, then evolution of stars and elements on the earth. Then we have the origin of earth. 4.6 billion years ago the earth originated, and then we had this hydrogen, water, ammonia, methane, and all these molecules got collided with each other and they formed into amino acids. And then we have proteins from this, then you have carbohydrates and lipids, then DNA. The DNA underwent self replication, then you have nucleoproteins. Nucleoproteins. So what happened here is, these nucleoproteins, nothing but proteins, means the nucleic acids were covered by the proteins, because nucleic acids are negatively charged, whereas the proteins are positively charged. So they form a colloidal system. They bind to each other. So then what happened, these are called as coacervates. They are called as coacervates. So these coacervates are called to be the first forms of life on earth. So, or then these coacervates, they gave rise to protobionts, protocells, or eobionts. Then from these protobionts, they started consuming the water around them, and they started undergoing nutrition. So when they started having the food, they got energy, and because of energy they were able to divide. So they started dividing. So that means nutrition, reproduction. So as you all have studied in the living world chapter, the very important characters of living organisms is, first is nutrition, and the second one is reproduction. So that was first seen in the protobionts. So these protobionts were able to have consume something and reproduce, and they gave rise to the first cells, that is the heterotrophs. Heterotrophs because they were not able to synthesize their own food, because there was no availability of chlorophyll. Chlorophyll was not available on the earth at that time. So therefore, they were dependent on other parts which were there surrounding. That is why we call them as heterotrophs. So some of the heterotrophs, what they did is, they started to use some chemicals in their body and trap the sunlight and prepare their own food. And that is what the chemoautotrophs or the autotrophs. So that is how the autotrophs existed, and these autotrophs gave rise. So once the plants were there, once the autotrophs were available on the earth, then the cooling was very faster, because these autotrophs, they took carbon dioxide and gave out oxygen. So because the availability of oxygen was more, the temperature of the earth started cooling very fast. So when the temperature started cooling very fast, now the clouds were formed because of transpiration. Okay, so now the life started existing on the earth. Okay, next, here we have the Miller's experiment. We have the Miller's experiment, that is, the Miller's experiment says that, you can see the Miller's experiment, correct? So here in the Miller's experiment, we can see there is a spark discharge chamber, there is a round bottom flask, there is a condenser, so there is a vacuum pump, and there is a U-trap tube. Okay, so this is what actually is an experiment which is very, very important for the exam for 5 marks or 3 marks. So what Miller did is, Miller took the different gases, that is hydrogen, ammonia and methane, in the ratio of hydrogen about 2% and ammonia about 1% and methane about 2%, mix that in the spark discharge chamber which were supported by the electrodes. Okay, then he connected all the vacuum pump was connected to this. Then you have a boiling water which is done in the round bottom flask. Okay, and then you have a U-trap tube, then a condenser. Now what Miller did is, the entire setup was stimulated to the primordial earth. So whatever was the primordial earth, sterile environment. Okay, so sterile environment was there, no life, no oxygen, nothing. So what did they do is, they started boiling the water was started boiling, then the vapor started moving through this vacuum, through this tube. It reached the spark discharge chamber. They mixed up with these gases. Then when they started condensing, they started condensing into this U-trap tube. So this experiment was conducted for about 18 days. After 18 days, they collected the sample and they saw that these gases had given rise to the amino acids like aspartic acid, alanine, and other components. So Miller's experiment is a very good proof to say chemical evolution has occurred. So we can now conclude that Earth has existed because of chemical evolution. Okay, so what is happening here is, this is a very, very important diagram which will be asked in the exam for 3 marks. Please don't forget it. Practice this diagram. Sorry. So you have a spark discharge chamber. You have a vacuum pump which will remove all the organisms in this tube. Then you have a round bottom flask. You have a U-trap tube and a condenser which will convert all the vapors into liquid. So that is the Miller's experiment, which proves chemical evolution, which says that yes, chemical evolution has occurred on the earth. So let us go on to the next slide. The next slide is about evidences. What are the evidences about evolution? So we have comparative morphology, we have embryology and paleontology, which gives evidence about the evolution has occurred. So when you talk about comparative morphology, so homologous organs and analogous organs, this is very, very, very important for the exam. There will be a question from this part. Homologous organs, these are the organs which have common origin but survive in different habitat with different function. So homologous organs means these are the organs which look very similar. I have an example. So you can see the forelimb of man, cheetah, whale, and bat. See, all look similar. They have the humerus bone, the tibia, fibula, all. But the man is using the forelimb for locomotion, the cheetah is using for running, the whale is using for flight, swimming, and the bat is using for flight. So that means all these organs have same structure, but the function is totally different. So that is called as homologous organs. And remember, homologous organs help in divergent evolution. They diverge the organism to different regions of the earth. Next, we have the analogous organs. Siddhagowda sir.
ಹೇಳಿ ಮೇಡಮ್.
ಸರ್, ಇದು ಕರೆಂಟ್ ಹೋಗಿದೆ ಇಲ್ಲಿ.
ಚೇಂಜ್ ಆಗಲ್ವಾ?
ಸ್ಲೈಡ್ ತರನಾಗುತ್ತೆ, ಕಂಪ್ಯೂಟರ್ ಆಫ್ ಆಗ್ಬೋದು ಅನ್ಸುತ್ತೆ ಮೋಸ್ಟ್ಲಿ.
ಅಲ್ಲ, ತೊಂದರೆ ಇಲ್ಲ. ಅಕಸ್ಮಾತ್ ನಿಮ್ಗೆ ನಿಮ್ಮ ಮಾಮೂಲಿ ನೀವು ಬೋರ್ಡ್ ಅಲ್ಲಿ ಮಾಡ್ತಿರಲ್ಲ, ಆತರ ಮಾಡ್ತೀನಿ ಅಂತಂದ್ರೆ ಸೊ ಮಾಡ್ತಾ ಇರಿ. ಕರೆಂಟ್ ಏನಾರ ಬಂದ್ರೆ ಕನೆಕ್ಟ್ ಆಗುತ್ತೆ, ಏನ್ ತೊಂದರೆ ಇಲ್ಲ.
ಹೌದಾ?
ಹಾ ಹಾ,
ಸರಿ ಸರ್, ಆಯ್ತು. ಓಕೆ ಓಕೆ. So analogous organs means these analogous organs, they have a different structure. The structure is totally different. You can see here, this is onion and this is opuntia. ಪಾಪಸ್ಕಳ್ಳಿ ಅಂತ ಹೇಳ್ತಾರಲ್ಲ ಕನ್ನಡದಲ್ಲಿ. ಸೊ ಇದು ಈರುಳ್ಳಿ, ಇದು ಪಾಪಸ್ಕಳ್ಳಿ. ನೋಡಿ, ಇದು ಈರುಳ್ಳಿಯ ಎಲೆಗಳು, ಇದು ಪಾಪಸ್ಕಳ್ಳಿ ಮೇಲಿರುವಂತ ಮುಳ್ಳುಗಳು. ಸಿ, ದಿಸ್ ಇಸ್ Sorry, this is also homologous organs. This is a plant example. This is an animal example. ಇಲ್ಲಿ ನೋಡಿ ಮಕ್ಕಳ, ಪ್ಲಾಂಟ್ ಅಲ್ಲಿ ಏನೋ ಇದು ಲೀಫೆ, ಈ ಮುಳ್ಳುಗಳು ಲೀಫೆ, ಬಟ್ ಆದ್ರೆ ಇಲ್ಲಿ ಸ್ಟೋರೇಜ್ ಹೆಲ್ಪ್ ಮಾಡ್ತಾ ಇದೆ, ಇಲ್ಲಿ ಡಿಫೆನ್ಸ್ ಮೆಕ್ಯಾನಿಸಮ್ ಹೆಲ್ಪ್ ಮಾಡ್ತಾ ಇದೆ. ಸೊ ದಿಸ್ ಇಸ್ ಆಲ್ಸೋ ಎಕ್ಸಾಂಪಲ್ ಫಾರ್ homologous organs. This is an animal example, this is a plant example. So please remember, very important for the exam, homologous organs, animal example is forelimb of man, cheetah, whale and bat, and plant example is scaly leaves of onion and spines of opuntia. So let us come to analogous organs. So if you see analogous organs, you can see the wing of the butterfly and wing of the bat. They are totally different, but both of them help in flight. So this is also helpful in flight and this is also helpful in flight. That means what? Because they share the same habitat, they live in the same sky, so therefore they have undergone the modification of flight. The structure is different, but that function is same. Here also you have potato and sweet potato. So potato is a stem modification, sweet potato is a root modification. This is the stem, this is the root, but still they undergo storage because the stem of the potato is underground, so it is modified for storage in function. So this is the example for analogous organs. So analogous organs means they have the same habitat, so therefore they perform the similar function. So organs are different, the organs are different, but they perform the same function because they share the same habitat. So these analogous organs give rise to convergent evolution. Convergent means they make the organisms to come together to share the same habitat. So that is what is called as analogous organs. So please remember the difference between homologous organs and analogous organs, and also remember the examples, plant example and animal example of both. Now let us come to evidences from embryology. So this is the evidence what you can get from embryology, where they say that you can see in the primordial life, I mean in the primitive life, all the embryos look similar. But if you see here, there are some changes which are taking place. But if you see here, they are totally changed. You can see how the changes have taken place. That means what? We were all same when we are in the embryonical stage, but we undergo differentiation during the developmental stages. So what that says? It says that we are developed from common ancestor. We were all zygotes, we were all unicellular organisms once in our lifetime, but now we have become into a very complex animal. That is, we all have developed from our ancestor. So there is one saying called as ontogeny. It recapitulates phylogeny. So ontogeny means our development, which recapitulates, which repeats our prehistory. So that means we say that our development shows that we will all come from our ancestors. So we were all once proto-amoeba, and how that amoeba has developed into a human. So that is what this diagram says. It says that we were all same in our embryonic stage, but as we started developing, we formed into fish, salamander, tortoise, and the chick. Okay, so that is one evidence to say evolution has occurred. Then we also have very important evidences from paleontology. What is paleontology? Study of fossils. What are fossils? So I had shown here in the previous slide what are this is a fossil. You can see there is a fossil here. This is a fossil of Archaeopteryx. It is a connecting link between a reptile and a bird. So this Archaeopteryx has the wings of a bird and it has the clawed legs as of a reptile. So it has both the characters of bird and a reptile. So this Archaeopteryx is a connecting link. It says that the reptiles have given rise to birds. So that is the evidences what you get from paleontology. So there are a lot of evidences which can link between the bird between different organisms. So you can see a imaginary picture of Archaeopteryx where it has beak as with teeth as that of a reptile. You can see wings and clawed legs as of a bird. So it resembles both of a bird and a reptile. So from fossils, we get a very, very important evidences that evolution has occurred in the earth, or evolution is occurring. Now coming on to theories of evolution, there are a lot of theories to talk about evolution. So you have the first one is Lamarckism. So this is a very, very ancient theory which was proposed by Lamarck, and it is popularly known as inheritance of acquired characters. So what is inheritance of acquired characters? Whatever the characters we get after we are born, we are going to keep acquiring it. So after you're born, we start shouting, we call our mother, we start walking, we start speaking, we start writing, we start cycling. So all these are nothing but the characters what we acquire during our lifetime. So Lamarck says that we acquire the characters by using our organs or by disusing our organs. So what are the postulates of Lamarck? He says two things. One is use theory and disuse theory. So very simple. Use means if you keep using your organs, if you keep using your brain, your heart, your lungs, your important organs, they become more and more efficient. So that is what we can see during the course of evolution from fishes to human. Fishes have less convolutions in the brain because they use less brain. They don't use their brains more when compared to humans. But when you come to frogs, little more convolutions are there. But when you come to reptiles, little more convolutions. But when you come to humans, highly convoluted brain. Convolutions means folds. The brain has so many folds. So folds indicate the IQ of the organism. So that shows that if an organ is used continuously, the efficiency of the organ increases. That is called as use theory. So that for that there is a very excellent example, that is the long neck of giraffe. So we all know that giraffe has the same cervical bones as we have, but because it is continuously using those neck bones, the neck of the giraffe has increased, because if it doesn't use the neck, it will die because of starvation. So it doesn't want to die, it wants to survive. So what has happened? The neck bones have got elongated, and that character is acquired from generation to generation, and then you have long-necked giraffe in the present generation. So that is a very good example for use theory. Then you have disuse theory. This is the photo of the Indian python, which shows the spur or the buds of the legs. ಇದನ್ನ ಕಾಲುಗಳು ಅಂತ ಕರೀತಾರೆ, ಕಾಲಿನ ಮೊಗ್ಗುಗಳಿದು ಸ್ಪರ್ ಅಂತಾರೆ ಮಕ್ಕಳ. ಸೊ ಇಲ್ಲಿ ಇಂಡಿಯನ್ ಪೈಥಾನ್ ಅಲ್ಲಿ ಸ್ಕೆಲಿಟನ್ ಅಲ್ಲೇ ಇದಿರುತ್ತೆ. ಅಂದ್ರೆ ಏನು? ಡಿಸ್ಯೂಸ್ ಪೈಥಾನ್ಸ್ ಅಥವಾ ಸ್ನೇಕ್ಸ್ ಅವರು ಉರುಳಾಡೋದಕ್ಕೆ ಈಸಿ ಪಡ್ತಾರೆ. ದೇ ವರ್ ವೆರಿ ಈಸಿ ಇನ್ ಕ್ರೀಪಿಂಗ್ ಅಂಡ್ ಕ್ರಾಲಿಂಗ್. they were feeling very difficult to use their legs to walk. So ಕಾಲಿಂದ ಓಡಾಡ್ತಾ ಇದ್ರೆ ಅವರು ಸಿಕ್ಕಾಕೊಂತ ಇದ್ರು, ಬಂದು ಬೇರೆಯವ್ರು ತಿಂತಾ ಇದ್ರು. So they were very easily able to creep or crawl. So because of that, what happened? The legs started degenerated. They became rudimentary. Rudimentary ಅಂದ್ರೆ ಅವು ಕೆಲಸ ಮಾಡ್ದಂಗಾಗಿ ಅವು ನಿಷ್ಕ್ರಿಯವಾಗೋಯ್ತು. ಆ ಕಾಲ್ಗಳು ನಿಷ್ಕ್ರಿಯವಾಗಿ ಅವು ಅವರ ಗಾತ್ರವನ್ನ ಕಳ್ಕೊಂಡ್ಬಿಟ್ರು. ದೇ ಡಿಕ್ರೀಸ್ ದೇರ್ ಸೈಜ್. ಸೊ ಅದನ್ನೇ ಸ್ಪರ್ ಅಂತ ಕರೀತಾರೆ. ಸ್ಪರ್ ಅಂದ್ರೆ ಲೆಗ್ ಬಡ್ಸ್ ಅಥವಾ ಲೆಗ್ ಬೋನ್ಸ್ ಅಂತ ಕರೀತಾರೆ, ರುಡಿಮೆಂಟರಿ ಲೆಗ್ ಬೋನ್ಸ್. ಸೊ ಇದೇನಂತ ಅರ್ಥ ಆಗ್ತಿದೆ? ನಾವ್ ಯಾವ ಅಂಗಾಂಗವನ್ನ ಉಪಯೋಗಿಸದಲ್ಲೇ ಇರ್ತೀವೋ ಆ ಅಂಗಾಂಗ ನಿಷ್ಕ್ರಿಯವಾಗ್ತಾ ಹೋಗುತ್ತೆ, ಅದು ಕಡಿಮೆ ಆಗ್ತಾ ಹೋಗುತ್ತೆ, ಅದರ ಕೆಲಸ ಕಡಿಮೆ ಆಗುತ್ತೆ, ಅದರ ಗಾತ್ರ ಕಡಿಮೆ ಆಗಿ ಅದು ನಮ್ಮ ದೇಹದಲ್ಲಿ ಸುಮ್ನೆ ಹಾಗೆ ವೇಸ್ಟ್ ಆಗಿ ಇರುತ್ತೆ. ಸೊ ಇದು ನಮ್ಮ ದೇಹದಲ್ಲಿ ತುಂಬಾ ಅಂಗಾಂಗಗಳಿದೆ ಮಕ್ಕಳ. ಕಾಕಾಯ್ ಅಂತ ಒಂದು ಬೋನ್ ಇದೆ. ನಮ್ಮ ವರ್ಟಿಬ್ರಲ್ ಕಾಲಮಿನ ಕೊನೆಯ ಬೋನ್ ಯಾವುದು? ಕಾಕಾಯ್. ಅದು ಕೂಡ ನಾವು ಬಾಲವನ್ನ ಯೂಸ್ ಮಾಡದಲ್ಲೇ ಏನಾಗಿದೆ, ಅದು ಒಂದು ಚಿಕ್ಕ ಮೂಳೆ ಬೋನ್ ಆಗಿ ಉಳ್ಕೊಂಡಿದೆ. ಹಾಗೆ ಸರ್ವಿಕ್ಸ್ ಅಂತ ಹೇಳ್ತಾರೆ. ಸರ್ವಿಕ್ಸು ಅಷ್ಟೇನೆ. ಮುಂಚೆ ನಮ್ಗೆ ಅದು ನಾವು ಯೂಸ್ ಮಾಡ್ತಾ ಇದ್ವಿ, ಅಂದ್ರೆ ಈ ಹಸುಗಳಲ್ಲಿ ಮತ್ತೆ ಬೇರೆ ಪ್ರಾಣಿಗಳಲ್ಲಿ ಅದು ದೊಡ್ಡದಾಗಿರುತ್ತೆ, ಯಾಕೆಂದ್ರೆ ಅವ್ರು ಜಾಸ್ತಿ ಹರ್ಬಿವರಸು. ಬಟ್ ನಾವು ಓಮ್ನಿವರಸ್ ಆಗಿರೋದ್ರಿಂದ ನಾವೇನಾಗ್ತಾ ಇದೆ ಅದು ನಮ್ಗೆ ಬೇಕಾಗಿಲ್ಲ. ಸೊ ಹಾಗಾಗಿ ಏನಾಗಿದೆ, ಅದನ್ನ ಯೂಸ್ ಮಾಡ್ದಲೆ ಅದು ಒಂದು ಚಿಕ್ಕ ಸ್ಯಾಕ್ ಆಗಿದೆ. ನಿಮ್ಗೆಲ್ಲರಿಗೂ ಗೊತ್ತು, ವರ್ಮಿಕ್ಸ್ ಅಂತ ಕರೀತಾರೆ ಅಥವಾ ಅಪೆಂಡಿಕ್ಸ್ ಅಂತ ಕರೀತಾರೆ. ಆ ಅಪೆಂಡಿಕ್ಸ್ ನಾವು ತೆಗ್ದಾಕ್ತಿವಿ ಅದು ಇನ್ಫೆಕ್ಷನ್ ಆದ್ರೆ. ಸೊ ಇವೆಲ್ಲವೂ ಮಕ್ಕಳ ಡಿಸ್ಯೂಸ್ ಥಿಯರಿ ಅಂದ್ರೆ ಏನೋ, ಯಾವ ಅಂಗಗಳನ್ನ ನಾವು ಯೂಸ್ ಮಾಡಲ್ವೋ ಅದು ನಮ್ಮ ದೇಹದಲ್ಲಿ ನಿಷ್ಕ್ರಿಯವಾಗ್ತಾ ಹೋಗುತ್ತೆ. ಅಂದ್ರೆ ಒಂದೇ ಜನರೇಷನ್ಗೆ ಆಗಲ್ಲ, ಎಷ್ಟೋ ಜನರೇಷನ್ಗಳಾದ ಮೇಲೆ ಅದು ನಿಷ್ಕ್ರಿಯವಾಗುತ್ತೆ. ಮಿಲಿಯನ್ಸ್ ಆಫ್ ಜನರೇಷನ್ ಆಗ್ಬೇಕು ಅದು ನಿಷ್ಕ್ರಿಯ ಆಗೋದಕ್ಕೆ. ಸೊ ದಟ್ ಈಸ್ ವಾಟ್ ದ ಥಿಯರಿ ಆಫ್ ಎವಲ್ಯೂಷನ್. ದ ಫಸ್ಟ್ ಥಿಯರಿ ಇಸ್ ಲೆಮಾರ್ಕಿಸಮ್. The next one is Darwinism. So this is, I think last week we had Charles Darwin Day. So ನಿಮ್ಗೆಲ್ಲರಿಗೂ ಇವತ್ತು ಎಲ್ಲಾರು ಸ್ಟೇಟಸ್ ಅಲ್ಲಿ ಡಾರ್ವಿನ್ ಫೋಟೋ ಹಾಕೊಂಡಿದ್ರು. ಸೊ ಡಾರ್ವಿನಿಸಮ್ ಏನು ಅಂದ್ರೆ, ಡಾರ್ವಿನ್ ಹ್ಯಾಡ್ ಎ ವೆರಿ ಗುಡ್ ಆಪರ್ಚುನಿಟಿ ಟು ಗೋ ಫಾರ್ ಅ ವಾಯೇಜ್ ಥ್ರೂ ಔಟ್ ದ ಅರ್ತ್. ಅಂದ್ರೆ ಇಡೀ ಭೂಮಿಯನ್ನ ಸುತ್ತೋದಕ್ಕೆ ಅವ್ರಿಗೆ ಒಂದು ಚಾನ್ಸ್ ಸಿಕ್ತು. ಅವರು ಬೀಗಲ್ ಅನ್ನೋ ಒಂದು ಶಿಪ್ ಅಲ್ಲಿ ಇಡೀ ಭೂಮಿಯನ್ನ ಸುತ್ತಿದ್ರು, ವಾಯೇಜ್ ಮಾಡಿದ್ರು. ಅವರಲ್ಲಿ ಒಬ್ರು ನ್ಯಾಚುರಲಿಸ್ಟ್ ಆಗಿ ಅವರು ಅಪಾಯಿಂಟ್ ಆಗಿದ್ರು. ಸೊ ಅವ್ರು ಏನ್ ಮಾಡ್ಬೇಕು ಅಂದ್ರೆ, ಸ್ಪೆಸಿಮೆನ್ಸ್ ನ ಕಲೆಕ್ಟ್ ಮಾಡ್ಬೇಕು, ಅದನ್ನ ಅವರು ಅನಲೈಸ್ ಮಾಡ್ಬೇಕು, ಅದರಿಂದ ಡೇಟಾ ಕಲೆಕ್ಟ್ ಮಾಡಿ ಅವರು ನ್ಯಾಚುರಲ್ ಸೊಸೈಟಿಗಳಿಗೆ ಸಬ್ಮಿಟ್ ಮಾಡ್ಬೇಕು. ಸೊ ಅವರು ಆತರ ಮಾಡಿದಾಗ ಒಂದು ಐಲ್ಯಾಂಡ್ ಹೋದ್ರು, ಯಾವುದು ಗೆಲಪಗಸ್ ಐಲ್ಯಾಂಡ್ ಅಂತ ಹೋದ್ರು. ಆ ಗೆಲಪಗಸ್ ಐಲ್ಯಾಂಡ್ ಹೋದಾಗ ಅಲ್ಲಿ ಅವ್ರು ನೋಡಿದ್ ಏನು ಅಂದ್ರೆ ಮಕ್ಳ, ಗೆಲಪಗಸ್ ಐಲ್ಯಾಂಡ್ ಅಲ್ಲಿ ಅವ್ರು ಏನ್ ನೋಡಿದ್ರು ಅಂದ್ರೆ, ಅಲ್ಲಿ ವಿಚಿತ್ರವಾದ ಜೀವಿಗಳು ಒಂದೇ ಕಡೆ ವಾಸ ಮಾಡ್ತಾ ಇದ್ವು. ಅಂದ್ರೆ ಬೇರೆ ಬೇರೆ ರೀತಿ ಜೀವಿಗಳು, ಒಂಥರ ಇರ್ಲಿಲ್ಲ, ಎಲ್ಲ ವಿಚಿತ್ರವಾಗಿರೋ ಜೀವಿಗಳನ್ನ ಅವ್ರು ನೋಡಿದ್ರು. ಆವಾಗ ಅವ್ರಿಗೆ ಮನ್ಸಲ್ಲಿ ಕೆಲವು ವಿಷಯಗಳ ಬಂತು. ಅದ್ ಯಾವ ವಿಷಯಗಳು ಅಂದ್ರೆ, ಕೆಲವು ಪಾಸ್ಟುಲೇಟ್ಸ್ ಬಂತು. ಓವರ್ ಪ್ರೊಡಕ್ಷನ್, ಸ್ಟ್ರಗಲ್ ಫಾರ್ ಎಕ್ಸಿಸ್ಟೆನ್ಸ್, ವೇರಿಯೇಷನ್ ಅಂಡ್ ಹೆರಿಡಿಟಿ, ಸರ್ವೈವಲ್ ಆಫ್ ದ ಫಿಟ್ಟೆಸ್ಟ್ ಬೈ ನ್ಯಾಚುರಲ್ ಸೆಲೆಕ್ಷನ್. ಸೊ ಏನು ಅಂದ್ರೆ, ಓವರ್ ಪ್ರೊಡಕ್ಷನ್ ಅಂದ್ರೆ ಏನು, ಅತಿಯಾಗಿ ಉತ್ಪನ್ನ ಮಾಡೋದು. ಯೂಶಲಿ ನೀವು ನೋಡಿ, ಎಲ್ಲಾ ಜೀವಿಗಳು ಅತಿಯಾಗಿ ಉತ್ಪನ್ನ ಮಾಡ್ತಾರೆ, ಆದ್ರೆ ಆ ಉತ್ಪನ್ನ ಮಾಡಿದಂತ ಎಲ್ಲಾ ಜೀವಿಗಳು ಭೂಮಿಯಲ್ಲಿ ಇರೋದಿಲ್ಲ, ನಶಿಸಿ ಹೋಗ್ತವೆ. ಸೊ ದಟ್ ಈಸ್ what is called as overproduction. For example, starfish na fastest breeder anta ಕರೀತಾರೆ. fastest breeder ಅಂದ್ರೆ ಬೇಗ ರೀಪ್ರೊಡ್ಯೂಸ್ ಮಾಡಿ ಬಿಡುತ್ತೆ. ಬಟ್ ಆದ್ರೆ ಜಗತ್ತಿನ ತುಂಬಾ ಸ್ಟಾರ್ಫಿಶ್ ಗಳಿಲ್ಲ. ಯಾಕೆ? ಆ ಸ್ಟಾರ್ಫಿಶ್ ಗಳನ್ನ ತಿನ್ನೋದಕ್ಕೆ ಅಂತಾನೆ ಬೇರೆ ಜೀವಿಗಳು ಹುಟ್ಕೊಂಡಿರ್ತಾರೆ. ಸೊ ದಟ್ ಈಸ್ ಓವರ್ ಪ್ರೊಡಕ್ಷನ್. ಸೊ ಎಷ್ಟೇ ಓವರ್ ಪ್ರೊಡಕ್ಷನ್ ಆದ್ರೂ ಮಕ್ಳ, ಅಲ್ಲಿ ಸ್ಟ್ರಗಲ್ ಫಾರ್ ಎಕ್ಸಿಸ್ಟೆನ್ಸ್ ಇರುತ್ತೆ. ಸ್ಟ್ರಗಲ್ ಅಂದ್ರೆ ಏನೋ, ದೇ ಕೀಪ್ ಸ್ಟ್ರಗ್ಲಿಂಗ್ ಫಾರ್ ದೇರ್ ಎಕ್ಸಿಸ್ಟೆನ್ಸ್, either for shelter or for food or for mating, for any one thing they'll be struggling. So ಆ ಸ್ಟ್ರಗಲ್ ಮಾಡ್ಬೇಕಾದ್ರೆ ಏನಾಗುತ್ತೆ, ವೇರಿಯೇಷನ್ ಬರುತ್ತೆ. ಅವರ ದೇಹದಲ್ಲಿ ಹಲವಾರು ಬದಲಾವಣೆಗಳು ಬರುತ್ತೆ, ಅವರ ಬಿಹೇವಿಯರ್ ಅಲ್ಲಿ ಹಲವಾರು ಬದಲಾವಣೆಗಳು ಬರುತ್ತೆ. ಅದನ್ನ ಅವರು ಕ್ಯಾಪ್ಚರ್ ಮಾಡ್ಕೊಂತಾರೆ, ಒಂದ್ ಜನರೇಷನ್ ಇಂದ ಒಂದ್ ಜನರೇಷನ್ಗೆ ಪಾಸ್ ಮಾಡ್ತಾರೆ. ಪಾಸ್ ಮಾಡಿದಾಗ ಯಾವ ಜೀವಿ ಬದುಕುತ್ತೆ ಅದನ್ನ ಸರ್ವೈವಲ್ ಆಫ್ ದ ಫಿಟ್ಟೆಸ್ಟ್ ಅಂತ ಕರೀತಾರೆ. ಹೇಗೆ? ನೇಚರ್ ಸೆಲೆಕ್ಟ್ ಮಾಡ್ಕೊಳುತ್ತೆ. ಐ ಆಮ್ ಸಾರಿ, ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ಹೇಳ್ಬೋದು ಕಾಕ್ರೋಚ್. ನಾನು ಯಾವಾಗ್ಲೂ ಈ ಎಕ್ಸಾಂಪಲ್ ಕೊಡ್ತೀನಿ. ಕಾಕ್ರೋಚ್ಗಳು ಮಕ್ಳ, ಕಾಕ್ರೋಚ್ಗಳು ಡೈನೋಸಾರ್ಗಳಿಗಿಂತ ಮುಂಚೆ ಇದ್ರು. ಬಟ್ ಆದ್ರೆ ಕಾಕ್ರೋಚ್ಗಳು ಇವಾಗ್ಲೂ ಕೂಡ ಸರ್ವೈವ್ ಆಗ್ತಿದ್ರೆ, ಡೈನೋಸಾರ್ ಸರ್ವೈವ್ ಆಗ್ತಿಲ್ಲ. ಕಾರಣ ಏನು ಅಂದ್ರೆ, ಕಾಕ್ರೋಚ್ಗಳಲ್ಲಿರೋ ಕೆಪಬಿಲಿಟಿ, ಅವ್ರ ಅಡಾಪ್ಟೇಷನ್, ಅವ್ರ ಕೈಟಿನಸ್ ಕವರಿಂಗ್. ಸೊ ಇದು ಒಂದು ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ ನಾನು ನ್ಯಾಷನಲ್ ಜಿಯೋಗ್ರಫಿಕ್ ಚಾನಲ್ ಅಲ್ಲಿ ನೋಡಿದ್ದೆ. ಏನು ಅಂದ್ರೆ, ಒಂದು ಟಿವಿ ಬಾಕ್ಸ್ ಒಳಗಡೆ ಒಂದಷ್ಟು ಕಾಕ್ರೋಚ್ಗಳು ಹಾಕ್ಬಿಟ್ಟು ಎಕ್ಸ್ಪ್ಲೋರ್ ಮಾಡ್ತಾರೆ, ಅಂದ್ರೆ ಎಕ್ಸ್ಪ್ಲೋಷನ್ ಮಾಡ್ತಾರೆ, ಅದನ್ನ ಹೊಡಿತಾರ, ಟಿವಿನ ಹೊಡೆದಾಗ ಆರಾಮಾಗಿ ಕಾಕ್ರೋಚ್ಗಳು ಆಚೆ ಬರುತ್ತೆ. ಅಂದ್ರೆ ಏನ್ ಅರ್ಥ, ಕಾಕ್ರೋಚ್ಗಳು ಎಲ್ಲಿ ಬೇಕಾದ್ರೂ ಹೇಗ್ ಬೇಕಾದ್ರೂ ಸರ್ವೈವ್ ಮಾಡ್ತಾರೆ, ಏನ್ ಬೇಕಾದ್ರೂ ತಿಂತಾರೆ. ಸೊ ದಟ್ ಈಸ್ ವಾಟ್ ಈಸ್ ವೆರಿ ಇಂಪಾರ್ಟೆಂಟ್, ಸರ್ವೈವಲ್ ಆಫ್ ದ ಫಿಟ್ಟೆಸ್ಟ್. ಹೂ ಇಸ್ ಗೋಯಿಂಗ್ ಟು ಸರ್ವೈವ್? ಯಾರು ಸರ್ವೈವ್ ಆಗ್ತಾರೋ ಅವ್ರನ್ನ ನೇಚರ್ ಸೆಲೆಕ್ಟ್ ಮಾಡ್ಕೊಳುತ್ತೆ, ಅವ್ರನ್ನ ನೆಕ್ಸ್ಟ್ ಕಂಟಿನ್ಯೂಟಿ ಮಾಡುತ್ತೆ. ಸೊ ದಿಸ್ ಇಸ್ ವೆರಿ ವೆರಿ ಇಂಪಾರ್ಟೆಂಟ್ ಅಬೌಟ್ ಡಾರ್ವಿನಿಸಮ್. ದಟ್ ಈಸ್ ಓವರ್ ಪ್ರೊಡಕ್ಷನ್, ಸ್ಟ್ರಗಲ್ ಫಾರ್ ಎಕ್ಸಿಸ್ಟೆನ್ಸ್, ವೇರಿಯೇಷನ್ ಅಂಡ್ ಹೆರಿಡಿಟಿ, ಸರ್ವೈವಲ್ ಆಫ್ ದ ಫಿಟ್ಟೆಸ್ಟ್ ಬೈ ನ್ಯಾಚುರಲ್ ಸೆಲೆಕ್ಷನ್. ಜ್ಞಾಪಕ ಇಟ್ಕೊಳ್ಳಿ, ಎಕ್ಸಾಮ್ ಅಲ್ಲಿ ಕೇಳ್ತಾರೆ. ನೆಕ್ಸ್ಟ್ ಇಂಪಾರ್ಟೆಂಟ್ ಏನು ಅಂದ್ರೆ ಅಡಾಪ್ಟಿವ್ ರೇಡಿಯೇಷನ್. ಏನ್ ಅಡಾಪ್ಟಿವ್ ರೇಡಿಯೇಷನ್ ಅಂದ್ರೆ, ಅಡಾಪ್ಟಿವ್ ರೇಡಿಯೇಷನ್ ಅಂದ್ರೆ ದ ಡೈವರ್ಜೆನ್ಸಿ ಆಫ್ ಎನ್ ಆರ್ಗಾನಿಸಮ್. ಅಂದ್ರೆ ಏನು? ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ಇವಾಗ ನೋಡಿ, ಈ ನಮ್ಮ ಸ್ಕಿನ್ ಕಲರ್ ನೋಡಿ ಹೇಗೆ ಅಡಾಪ್ಟ್ ಆಗಿದೆ. ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ಈಗ ನಮ್ಮ ಇಂಡಿಯಾನಲ್ಲಿ ವೀಟ್ ಕಲರ್ ಸ್ಕಿನ್ ಇರುತ್ತೆ, ಅದೇ ಆಫ್ರಿಕಾಗ್ ಹೋದ್ರೆ ನೀಗ್ರೋ ಬ್ಲಾಕ್ ಕಲರ್ ಇರುತ್ತೆ, ಅದೇ ಅಮೆರಿಕಾಗ್ ಹೋದ್ರೆ ಫುಲ್ ವೈಟ್ ಇರ್ತಾರೆ. ಯಾಕೆ? ಅವ್ರು ಇರುವಂತ ಒಂದು ಟೆಂಪರೇಚರು. ಸೊ ಆ ಟೆಂಪರೇಚರ್ಗೆ ಅವ್ರ ಆ ಕಲರ್ ಇಲ್ಲ ಆದ್ರೆ ಅವರು ಆಗಲ್ಲ. ಸೊ ಅದೇ ಅಡಾಪ್ಟಿವ್ ರೇಡಿಯೇಷನ್. ಅಡಾಪ್ಟಿವ್ ರೇಡಿಯೇಷನ್ ಅಂದ್ರೆ ದ ಡೈವರ್ಜೆನ್ಸ್ ಆಫ್ ಎನ್ ಆರ್ಗಾನಿಸಮ್ ಟು ದ ನ್ಯೂ ಜಿಯೋಗ್ರಫಿಕಲ್ ಏರಿಯಾ ಬೇಸ್ಡ್ ಆನ್ ದ ಅವೈಲಬಿಲಿಟಿ ಆಫ್ ನ್ಯಾಚುರಲ್ ರಿಸೋರ್ಸಸ್. ಎಷ್ಟು ನ್ಯಾಚುರಲ್ ರಿಸೋರ್ಸಸ್ ಅವೈಲಬಲ್ ಇರುತ್ತೋ ಆ ಬೇಸಿಸ್ ಮೇಲೆ ಹೇಗೆ ಆರ್ಗಾನಿಸಮ್ಸ್ ಡೆವಲಪ್ ಆಗುತ್ತೆ. ಸೊ ದಿಸ್ ಇಸ್ ಅ ವೆರಿ ಇಂಪಾರ್ಟೆಂಟ್ ಕ್ವೆಶನ್ ವಿಚ್ ವಿಲ್ ಬಿ ಆಸ್ಕ್ ಫಾರ್ ಎಕ್ಸಾಮ್. ವೆರಿ ವೆರಿ ಇಂಪಾರ್ಟೆಂಟ್. ಇಲ್ಲಿ ನೋಡಿ, ಇದು ಡಾರ್ವಿನ್ ಫಿಂಚಸ್. ನಾನು ಹೇಳಿದ್ನಲ್ಲ, ಗ್ಯಾಲಪಗಸ್ ಐಲ್ಯಾಂಡ್ ಅಲ್ಲಿ ಡಾರ್ವಿನ್ ಇದನ್ನೇ ಕಂಡಿಡಿದಿದ್ದು. ಒಂದು ಫಿಂಚ್ ಇತ್ತು, ಇದು ಆನ್ಸೆಸ್ಟ್ರಲ್ ಫಿಂಚ್. ಬಟ್ ಆದ್ರೆ ಅವ್ನ್ ಎಲ್ಲಾ ಕಡೆ ನೋಡಿದಾಗ ಇದೇ ಫಿಂಚ್ ಇರ್ಲಿಲ್ಲ. ಕೆಲವು ಕಡೆ ಲಾರ್ಜ್ ಗ್ರೌಂಡ್ ಫಿಂಚ್ ಇತ್ತು, ಕೆಲವು ಕಡೆ ಕ್ಯಾಕ್ಟಸ್ ತಿನ್ನುವಂತ ಫಿಂಚ್ಗಳಿತ್ತು, ಕೆಳಗಡೆ ವಾರ್ಬಲ್ ತಿನ್ನುವಂತ ಫಿಂಚ್ಗಳಿತ್ತು, ಕೆಲವು ಕಡೆ ವುಡ್ ಪೆಕರ್ ಫಿಂಚಸ್ ಇತ್ತು, ಕೆಲವು ಕಡೆ ಟ್ರೀ ಫಿಂಚಸ್ ಇತ್ತು, ಕೆಲವು ಕಡೆ ಈ ತರ. ಸೊ ಅಂದ್ರೆ ಏನು, ಫ್ರೂಟ್ ಈಟರ್ಸ್, ಇನ್ಸೆಕ್ಟ್ ಈಟರ್ಸ್, ಕ್ಯಾಕ್ಟಸ್ ಈಟರ್ಸ್ ಮತ್ತೆ ಸೀಡ್ ಈಟರ್ಸ್. ಅಂದ್ರೆ ಅರ್ಥ ಏನು ಅಂದ್ರೆ, ಈ ಫಿಂಚಸ್ ಗಳು ತಮ್ಮ ಬೀಕ್ಗಳನ್ನ ಬದಲಾಯಿಸಿದ್ದಾರೆ. ಯಾಕೆ ಬದಲಾಯಿಸಿದ್ದಾರೆ ಬೀಕ್ಗಳನ್ನ? ಯಾವ ಆಹಾರವನ್ನ ಅವರು ತಿಂತಾರೋ ಅದ್ರ ತಕ್ಕಂಗೆ ಅವರು ತಮ್ಮ ಬೀಕ್ಗಳನ್ನ ಬದಲಾಯಿಸ್ಕೊಂಡಿದ್ದಾರೆ. ಸೊ ಯಾವ ಫಿಂಚಸ್ ಸೀಡ್ ನ ತಿನ್ಬೇಕು, ಅದನ್ನ ಬ್ರೇಕ್ ಮಾಡ್ಬೇಕು, ಅದಕ್ಕೆ ಶಾರ್ಪ್ ಇದೆ ಅವ್ರಿಗೆ ಬೀಕು. ಇಲ್ಲಿ ಕ್ಯಾಕ್ಟಸ್ ನ ತಿನ್ಬೇಕು, ಕ್ಯಾಕ್ಟಸ್ ನ ಬಗ್ದು ಅದನ್ನ ತಿನ್ಬೇಕು, ಅದಕ್ಕೆ ಅವ್ರಿಗೆ ಕ್ಯಾಕ್ಟಸ್ ಬೀಕ್ ಈ ತರ ಇದೆ. ಇದು ಇನ್ಸೆಕ್ಟ್ ಈಟರ್ಸ್, ಇನ್ಸೆಕ್ಟ್ ನ ಕುಕ್ಕಿ ಸಾಯಿಸ್ಬೇಕಲ್ಲ, ಸೊ ಅದಕ್ಕೆ ಫಿಂಚ್ ಏನೋ ಬೀಕು ಶಾರ್ಪ್ ಆಗಿದೆ. ಇಲ್ಲಿ ನೋಡಿ, ಫ್ರೂಟ್ ಈಟ್ ಪ್ಯಾಟರ್ಸ್, ಫ್ರೂಟ್ ನ ಶೆಲ್ ನ ಹೊಡಿಬೇಕು, ಸೊ ಅದಕ್ಕೆ ಈ ತರ ಸ್ವಲ್ಪ ಹುಕ್ಕಡ್ ಆಗಿದೆ. ಸೊ ಯಾವ ಫುಡ್ ನ ತಿಂತಾರೆ, ವಾಟ್ ದೇ ಈಟ್ will change the beak morphology in case of Darwin finches. So ಇದನ್ನ ಎಕ್ಸಾಮ್ ಅಲ್ಲಿ ಇಂಪಾರ್ಟೆಂಟ್ ಮಕ್ಕಳ ಏನ್ ಕೇಳ್ತಾರೆ ಅಂದ್ರೆ, ಎಕ್ಸ್ಪ್ಲೈನ್ ಅಡಾಪ್ಟಿವ್ ರೇಡಿಯೇಷನ್ ವಿತ್ ರೆಸ್ಪೆಕ್ಟ್ ಟು ಡಾರ್ವಿನ್ ಫಿಂಚಸ್ ಅಂತ ಕೇಳ್ತಾರೆ. ಸೊ ಇದನ್ನ ಗಮನ ಇಟ್ಟು ಓದಿ ಎಕ್ಸಾಮ್ಗೆ. ನೆಕ್ಸ್ಟ್, ಹಾರ್ಡಿ-ವಿನ್ಬರ್ಗಸ್ ಪ್ರಿನ್ಸಿಪಲ್. ಏನ್ ಹಾರ್ಡಿ-ವಿನ್ಬರ್ಗಸ್ ಪ್ರಿನ್ಸಿಪಲ್ ಅಂದ್ರೆ, ಹಾರ್ಡಿ-ವಿನ್ಬರ್ಗಸ್ ಲಾ ಅಂತ ಹೇಳ್ತಾರೆ, ಅಥವಾ ಹಾರ್ಡಿ-ವಿನ್ಬರ್ಗಸ್ ಲಾ ಆಫ್ ಈಕ್ವಿಲಿಬ್ರಿಯಂ ಅಂತ ಕರೀತಾರೆ. ಏನು ಹಾರ್ಡಿ-ವಿನ್ಬರ್ಗಸ್ ಲಾ ಆಫ್ ಈಕ್ವಿಲಿಬ್ರಿಯಂ ಅಂದ್ರೆ, ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ಇದನ್ನ ಇಲ್ಲಿ ಹೇಳ್ತೀನಿ ಇದನ್ನ. ಸೊ ಯು ಕ್ಯಾನ್ ಸಿ ಹಿಯರ್, in population of sexually reproducing organism in which random mating takes place, the frequency of particular genes and genotypes remain constant through generation of generation unless it is upset by elemental force of evolution. ಅಂದ್ರೆ ಅರ್ಥ ಏನು, ಒಂದು ಜನರೇಷನ್ ಇಂದ ಇನ್ನೊಂದು ಜನರೇಷನ್ ವರ್ಗು ಕ್ಯಾರೆಕ್ಟರ್ಸ್ ಹಾಗೆ ಪಾಸ್ ಆಗ್ತಾ ಇರುತ್ತೆ. ಎಲ್ಲಿವರೆಗೂ ಯಾವುದೇ ಒಂದು ಹೊರಗಡೆಯಿಂದ ಬಂದಿರೋ ಫ್ಯಾಕ್ಟರ್ಗಳ ಮೇಲೆ ಇನ್ಫ್ಲುಯೆನ್ಸ್ ಮಾಡ್ಲಿಲ್ಲ ಅಂದ್ರೆ ಆ ಕ್ಯಾರೆಕ್ಟರ್ ಒಬ್ರಿಂದ ಒಬ್ರಿಗೆ ಪಾಸ್ ಆಗ್ತಾ ಹೋಗುತ್ತೆ. ಅಂದ್ರೆ ಏನ್ ಅರ್ಥ ಅಂದ್ರೆ ಮಕ್ಳ, ಈಗ ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ಲಾಂಗ್ ನೆಕ್ ಜಿರಾಫೆ ತಗೊಳ್ಳಿ. ಅಲ್ಲಿ ಲಾಂಗ್ ನೆಕ್ ಜಿರಾಫೆ ಯಾಕೆ ಬಂದಿರೋದಂದ್ರೆ, ಆ ಕ್ಯಾರೆಕ್ಟರ್ ಪಾಸ್ ಆಗಿದೆ ಒಂದು ಜನರೇಷನ್ ಇಂದ ಇನ್ನೊಂದು ಜನರೇಷನ್ಗೆ, ಮತ್ತೊಂದು ಜನರೇಷನ್ಗೆ ಹಾಗೆ ಪಾಸ್ ಆಗಿರೋದ್ರಿಂದ ಅದು ಲಾಂಗ್ ನೆಕ್ ಜಿರಾಫೆ ಆಗಿದೆ. ಓಕೆ, ಸೊ ಅಂದ್ರೆ ಏನು, ಕ್ಯಾರೆಕ್ಟರ್ಸ್ ಒಬ್ರಿಂದ ಒಬ್ರಿಗೆ ಪಾಸ್ ಆಗ್ತಾ ಇದ್ರೆ ಅದು ಏನಾಗುತ್ತೆ, ಅದು ನಾರ್ಮಲ್ ಆಗಿರುತ್ತೆ. ಬಟ್ ಯೂಶಲಿ ಏನಾಗುತ್ತೆ, ಎವಲ್ಯೂಷನ್ ಆ ಕಾನ್ಸ್ಟೆಂಟ್ ಇದ್ರೆ, ನೋಡಿ ಇದು ಇಂಪಾರ್ಟೆಂಟ್ ಮಕ್ಳ. ಹಾರ್ಡಿ-ವಿನ್ಬರ್ಗಸ್ ಪ್ರಿನ್ಸಿಪಲ್ ಏನ್ ಅಂದ್ರೆ, p² + 2pq + q² = 1. ಏನು p² ಅಂದ್ರೆ, ಒಂದು ಪರ್ಟಿಕ್ಯುಲರ್ ಜಿನೋಟೈಪ್. ಓಕೆ, ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ಶಾರ್ಟ್ ನೆಕ್ಡ್ ಜಿರಾಫೆ ಅನ್ಕೊಳ್ಳಿ, ಇದು q² ಅಂದ್ರೆ ಲಾಂಗ್ ನೆಕ್ಡ್ ಜಿರಾಫೆ. p ಮತ್ತೆ q ಅಂದ್ರೆ ಎರಡು ಕ್ಯಾರೆಕ್ಟರ್, ಅವೆರಡು ಸೇರಿ ಯಾವಾಗ್ಲೂ ಕಾನ್ಸ್ಟೆಂಟ್ ಆಗಿರುತ್ತೆ. ಅಂದ್ರೆ ಏನರ್ಥ ಅಂದ್ರೆ, ಅವ್ರ ಹಾರ್ಡಿ-ವಿನ್ಬರ್ಗಸ್ ಲಾ ಆಫ್ ಈಕ್ವಿಲಿಬ್ರಿಯಂ ಅಂದ್ರೆ ಏನ್ ಹೇಳೋದು ಅಂದ್ರೆ, ಒಂದು ಕ್ಯಾರೆಕ್ಟರ್ ಒಬ್ರಿಂದ ಒಬ್ರಿಗೆ ಪಾಸ್ ಆಗ್ತಾ ಅದು ಕಾನ್ಸ್ಟೆಂಟ್ ಆಗಿ ಪಾಸ್ ಆಗ್ತಾ ಇರುತ್ತೆ. ಎಲ್ಲಿವರೆಗೂ ಕಾನ್ಸ್ಟೆಂಟ್ ಆಗಿ ಪಾಸ್ ಆಗಿರುತ್ತೆ, ಅದ್ರ ಮೇಲೆ ಯಾವ್ದಾದ್ರೂ ಹೊರಗಡೆಯಿಂದ ಕ್ಯಾರೆಕ್ಟರ್ ಪ್ರಭಾವ ಬೀರ್ಲಿಲ್ಲ ಅಂದ್ರೆ ಅದು ಕಾನ್ಸ್ಟೆಂಟ್ ಆಗಿ ಪಾಸ್ ಆಗ್ತಾ ಇರುತ್ತೆ. ಬಟ್ ಹೊರಗಡೆಯಿಂದ ಯಾವ್ದಾರ ಪ್ರಭಾವ ಬೀರಿದ್ರೆ ಯಾವ್ದು ಬೀಳ್ಬೋದು, ಇಲ್ಲಿದೆ ನೋಡಿ ಫ್ಯಾಕ್ಟರ್ಸ್. ನೋಡಿ, these are the factors which affect Hardy-Weinberg's law: gene mutation, gene migration, genetic drift, genetic recombination ಮತ್ತೆ ನ್ಯಾಚುರಲ್ ಸೆಲೆಕ್ಷನ್. ಜೀನ್ ಮ್ಯೂಟೇಶನ್ ಅಂದ್ರೇನು, ಹೊಸ ಜೀನ್ಸ್ ಗಳೇನಾರ ಫಾರ್ಮೇಶನ್ ಆದ್ರೆ ಆಗ ಈ ಕ್ಯಾರೆಕ್ಟರ್ ಮುಂದಕ್ಕೆ ಹೋಗಲ್ಲ, ಬೇರೆ ಕ್ಯಾರೆಕ್ಟರ್ ಹೋಗುತ್ತೆ, ಅವಾಗ ಹಾರ್ಡಿ-ವಿನ್ಬರ್ಗಸ್ ಲಾ ವರ್ಕ್ ಆಗಲ್ಲ. ಸೊ ಜೀನ್ ಮ್ಯೂಟೇಶನ್ ಗಳಾದ್ರೆ, ಅಂದ್ರೆ ನಮ್ಮ ದೇಹದ ಕ್ಯಾರೆಕ್ಟರ್ ಅಲ್ಲಿ ಬದಲಾವಣೆಗಳಾದ್ರೆ ಆಗ ಹಳೆ ಕ್ಯಾರೆಕ್ಟರ್ ಹಾಗೆ ಉಳಿಯುತ್ತೆ, ಹೊಸ ಕ್ಯಾರೆಕ್ಟರ್ ಬರುತ್ತೆ, ಆಗ ಈಕ್ವಿಲಿಬ್ರಿಯಂ ಹೋಗಲ್ಲ, ವರ್ಕೌಟ್ ಆಗಲ್ಲ, ಅದು ಕಾನ್ಸ್ಟೆಂಟ್ ಇರಲ್ಲ, ಚೇಂಜ್ ಆಗುತ್ತೆ. ಜೀನ್ ಮೈಗ್ರೇಷನ್ ಅಂದ್ರೇನು, ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ಈಗ ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ನಿಮ್ಗೆಲ್ಲರಿಗೂ ಗೊತ್ತು ಟರ್ಕಿನಲ್ಲಿ ಅರ್ಥಕ್ವೇಕ್ಸ್ ಆಗ್ತಾ ಇದೆ. ಸೊ ಅಲ್ಲಿ ಏನ್ ಮಾಡ್ಕೊಂಡಿದ್ದಾರೆ ಜನಗಳು, ಭಾರತದ ಜನಗಳು ಅಲ್ಲಿ ಗಿರಣಿ ಹಾಕಿದ್ರಂತೆ, ಅವರೆಲ್ಲ ಏನಾಗ್ತದೆ ಇಲ್ಲಿಗೆ ಬರ್ತಾರೆ ವಾಪಸ್ ದೇಶಕ್ಕೆ. ಸೊ ಏನಾಯ್ತು, ಜೀನ್ ಮೈಗ್ರೇಷನ್ ಆಯ್ತು, ಅಲ್ಲಿಂದ ಜನಗಳು ಇಲ್ಲಿಗೆ ಬರ್ತಾ ಇದಾರೆ, ಅಲ್ಲಿಂದ ಜೀನ್ಸ್ ಇಲ್ಲಿಗೆ ಬರ್ತಾ ಇದೆ. ಸೊ ಜೀನ್ ಫ್ಲೋ ಆದ್ರೆ, ಜೀನ್ಸ್ ಒಂದ್ ಪಾಪ್ಯುಲೇಷನ್ ಇಂದ ಇನ್ನೊಂದು ಪಾಪ್ಯುಲೇಷನ್ ಬಂದ್ರೆ ಆಗ್ಲೂ ಕೂಡ ಈ ಹಾರ್ಡಿ ವಿನ್ಬರ್ಗರ್ಸ್ ಈಕ್ವಿಲಿಬ್ರಿಯಂ ವರ್ಕ್ ಆಗಲ್ಲ. ಓಕೆ, ಜೆನೆಟಿಕ್ ಡ್ರಿಫ್ಟ್ ಅಂದ್ರೆ, ಡ್ರಿಫ್ಟ್ ಆದ್ರೆ ಆ ಚಿಕ್ಕ ಚಿಕ್ಕ ಪಾಪ್ಯುಲೇಷನ್ ಅಲ್ಲಿ ಏನಾರ ಬದಲಾವಣೆಗಳಾದ್ರೆ, ಏನಾರ ಡ್ರಿಫ್ಟ್ ಆದ್ರೆ, ಈಗ ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ಚಿಕ್ಕ ಚಿಕ್ಕ ಪಾಪ್ಯುಲೇಷನ್ಸ್ ಇರುತ್ತೆ, ಹಳ್ಳಿಗಳಲ್ಲಿ ಚಿಕ್ಕ ಚಿಕ್ಕ ಪಾಪ್ಯುಲೇಷನ್ಸ್ ಇರುತ್ತೆ, ಮಧ್ಯದಲ್ಲಿ ಫ್ಲಡ್ಡಿಂಗ್ ಆಯ್ತು, ಡ್ರಾಟ್ ಆಯ್ತು, ಅವಾಗ ಏನಾಗುತ್ತೆ, ಎಲ್ಲ ಏನಾಗ್ತಾರೆ, ಡೈವರ್ಜೆಂಟ್ ಆಗ್ಬಿಡ್ತಾರೆ, ಎಲ್ಲ ಬೇರೆ ಬೇರೆ ಕಡೆ ಹೋಗ್ತಾರೆ, ಚದ್ರಿ ಹೋಗ್ತಾರೆ. ಸೊ ಜೆನೆಟಿಕ್ ಡ್ರಿಫ್ಟ್ ಅಂದ್ರೇನು, ಫ್ಲಕ್ಚುಯೇಷನ್ ಇನ್ ಜೀನ್ ಫ್ರೀಕ್ವೆನ್ಸಿ ಇನ್ ಸ್ಮಾಲ್ ಪಾಪ್ಯುಲೇಷನ್. ಅವಾಗ ಏನಾಗ್ತಾರೆ, ಆ ಹಳ್ಳಿನಲ್ಲಿರೋ ಜನ ಬೇರೆ ಒಂದ್ ಹಳ್ಳಿಗೆ ಹೋಗ್ತಾರೆ, ಅಲ್ಲಿ ಅವ್ರಿಗೆ ಹೊಸ ವಾತಾವರಣ ಇರುತ್ತೆ, ಹೊಸ ಜಾಗ ಇರುತ್ತೆ, ಹೊಸ ನೀರು ಎಲ್ಲ, ಅದಕ್ಕೆ ಅವ್ರು ಅಡ್ಜಸ್ಟ್ ಆಗ್ಬೇಕು. ಅದಕ್ಕೆ ಅವಾಗ ಅವ್ರು ಅಲ್ಲಿಯ ಪಾಪ್ಯುಲೇಷನ್ ಆಗ್ತಾರೆ. ಆ ಒಂದು ಇದನ್ನ ಎಫೆಕ್ಟ್ ನ ಫೌಂಡರ್ಸ್ ಎಫೆಕ್ಟ್ ಅಂತ ಕರೀತಾರೆ ಮಕ್ಳ. ಇದು ತುಂಬಾ ಇಂಪಾರ್ಟೆಂಟ್. MCQ ಕ್ವೆಶನ್ಸ್ ಅಲ್ಲಿ ಕೇಳುವಂತ ಪ್ರಶ್ನೆ, ಫೌಂಡರ್ಸ್ ಎಫೆಕ್ಟ್. ಏನು ಫೌಂಡರ್ಸ್ ಎಫೆಕ್ಟ್ ಅಂದ್ರೆ, ಇಟ್ ಈಸ್ ನಥಿಂಗ್ ಬಟ್ due to fluctuation of the gene frequency in small populations, the population moves into a new region with new resources, with new habitat. So that population becomes a new population or the original population of the new habitat. ಆ ಹೊಸ ಜಾಗಕ್ಕೆ ಅವರು ಒಂದು ಹೊಸ ಪಾಪ್ಯುಲೇಷನ್ ಆಗ್ತಾರೆ, ಅವರು ಹೊಸದಾದ ಒಂದು ವಸ್ತುವನ್ನ ಕಂಡುಹಿಡಿತಾರೆ, ಅದಕ್ಕೆ ಅದನ್ನ ಫೌಂಡರ್ಸ್ ಎಫೆಕ್ಟ್ ಅಂತ ಕರೀತಾರೆ. ನೆಕ್ಸ್ಟ್, ಜೆನೆಟಿಕ್ ರಿಕಾಂಬಿನೇಷನ್. ಏನು ಜೆನೆಟಿಕ್ ರಿಕಾಂಬಿನೇಷನ್? ಇದು ಕೇವಲ ಸೆಕ್ಷುಯಲ್ ರಿಪ್ರೊಡಕ್ಷನ್ ಆಗುವಂತ ಜೀವಿಗಳಲ್ಲಿ ಮಾತ್ರ ಕಾಣೋದು ಮಕ್ಳ. ಇಲ್ಲೇನಾಗುತ್ತೆ ಅಂದ್ರೆ ಹೊಸ ಹೊಸ ಜೀವಿಗಳು ಹುಟ್ಟತಾನೆ ಇರ್ತವೆ. ನೀವ್ ನೋಡಿದ್ರೆ ಬಟರ್ಫ್ಲೈಸ್, ಎಷ್ಟೊಂಥರ ಬಟರ್ಫ್ಲೈಸ್ ಇದೆ, ಎಷ್ಟೊಂಥರ ಜೀವಿಗಳಿದೆ. ಒಂದ್ ನಿಮಿಷ. Okay, so due to sexual reproduction of offsprings are varied from parents resulting in evolution. ಹೊಸ ತರ ಜೀವಿಗಳು ಹುಟ್ಟುಕೊಳ್ಳೋದ್ರಿಂದ, ಈಗ ನಾವೇ ನೋಡಿ, ನಾವು ನಮ್ಮ ತಂದೆ ತಾಯಿ ತರ ಕೇವಲ 25% ಮಾತ್ರ ಡಿಫ್ರೆಂಟ್ ಇರ್ತೀವಿ, ಅವ್ರ ತರ ಜೆರಾಕ್ಸ್ ಆಗಿರೋಕ್ಕಾಗಲ್ಲ. ಸೊ ದಟ್ ಈಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಜೆನೆಟಿಕ್ ರಿಕಾಂಬಿನೇಷನ್. ಸೊ ಈ ತರ ಹೊಸ ಹೊಸ ಜೀವಿಗಳು ಹುಟ್ಕೊಂಡ್ರೆ ಹಾರ್ಡಿ ವಿನ್ಬರ್ಗರ್ಸ್ ಲಾ ಆಫ್ ಇಕ್ವಿಲಿಬ್ರಿಯಂ ಆಗೋದಿಲ್ಲ, ಅದು ಕಾನ್ಸ್ಟೆಂಟ್ ಇರೋದಿಲ್ಲ, ಚೇಂಜ್ ಆಗುತ್ತೆ, ಬದಲಾಗುತ್ತೆ. ನೆಕ್ಸ್ಟ್, ನ್ಯಾಚುರಲ್ ಸೆಲೆಕ್ಷನ್. ನ್ಯಾಚುರಲ್ ಸೆಲೆಕ್ಷನ್ ಅಂದ್ರೇನು? ಈ ಬದಲಾಗಿರೋ ಜೀವಿಗಳನ್ನ ನೇಚರ್ ಪಿಕ್ ಮಾಡುತ್ತೆ, ಪಿಕ್ ಮಾಡಿ ಅವ್ರನ್ನ ಮತ್ತೆ ರೀಪ್ರೊಡ್ಯೂಸ್ ಮಾಡಿ ಅವುಗಳ ಪಾಪ್ಯುಲೇಷನ್ ಹೆಚ್ಚಿಸುತ್ತೆ. ಸೊ ಅದರಿಂದನೂ ಕೂಡ ಹಾರ್ಡಿ ವಿನ್ಬರ್ಗರ್ಸ್ ಲಾ ಆಫ್ ಇಕ್ವಿಲಿಬ್ರಿಯಂ ಚೇಂಜ್ ಆಗುತ್ತೆ. ಮತ್ತೆ ಹೇಳ್ತಾ ಇದೀನಿ ಮಕ್ಳ, ಹಾರ್ಡಿ ವಿನ್ಬರ್ಗರ್ಸ್ ಪ್ರಿನ್ಸಿಪಲ್ ಎಕ್ಸಾಮ್ಗೆ ತುಂಬಾನೇ ಇಂಪಾರ್ಟೆಂಟ್. ನಿಮ್ಮ ಟೆಕ್ಸ್ಟ್ ಬುಕ್ ಅಲ್ಲಿ ಇದು ಡೆಫಿನೇಷನ್ ಕೊಟ್ಟಿದ್ದಾರೆ, ಇದನ್ನ ಬೇಕಾದ್ರೆ ನೀವು ಆಲ್ಟರ್ ಮಾಡಿ ಬರೀಬಹುದು. ಅಂದ್ರೆ ಏನೋ, in a population of sexually reproduced organism, the frequencies remain constant from generation to generation until they are forced by an external effect. ಅಂದ್ರೆ ಯಾವ್ದಾರ ಒಂದು ಎಕ್ಸ್ಟರ್ನಲ್ ಫೋರ್ಸ್ ಅದ್ರ ಮೇಲೆ ಎಫೆಕ್ಟ್ ಆದಾಗ ಅದು ಕಾನ್ಸ್ಟೆನ್ಸಿ ಬ್ರೇಕ್ ಆಗುತ್ತೆ. ಸೊ ಅದನ್ನ ಹಾರ್ಡಿ ವಿನ್ಬರ್ಗರ್ಸ್ ಲಾ ಅಂತಾರೆ. ಅದರ ಫ್ಯಾಕ್ಟರ್ಸ್ ಯಾವ್ಯಾವ್ದು ಅಂದ್ರೆ, ಜೀನ್ ಮ್ಯೂಟೇಶನ್, ಜೀನ್ ಮೈಗ್ರೇಷನ್, ಜೆನೆಟಿಕ್ ಡ್ರಿಫ್ಟ್, ಜೆನೆಟಿಕ್ ರಿಕಾಂಬಿನೇಷನ್ ಮತ್ತೆ ನ್ಯಾಚುರಲ್ ಸೆಲೆಕ್ಷನ್. ಎಕ್ಸಾಮ್ ಓದ್ಕೊಂಡೋಗಿ, ಅದು ಇಂಪಾರ್ಟೆಂಟ್. ಇನ್ನೊಂದು ಇಂಪಾರ್ಟೆಂಟ್ ಕ್ವಶನ್ ಯಾವ್ದಪ್ಪ ಅಂದ್ರೆ, ಇಂಡಸ್ಟ್ರಿಯಲ್ ಮೆಲಾನಿಸಮ್. ಏನ್ ಇಂಡಸ್ಟ್ರಿಯಲ್ ಮೆಲಾನಿಸಮ್ ಅಂದ್ರೆ, ಇಂಗ್ಲೆಂಡ್ ಅಲ್ಲಿ 1850 ನಲ್ಲಿ ಇಂಡಸ್ಟ್ರಿಯಲೈಸೇಷನ್ ಸ್ಟಾರ್ಟ್ ಆಯ್ತು, ಕಾರ್ಖಾನೆಗಳು ಸ್ಟಾರ್ಟ್ ಆದ್ರು. ಸೊ ಕಾರ್ಖಾನೆಗಳು ಸ್ಟಾರ್ಟ್ ಆಗೋಕ್ಕಿಂತ ಮುಂಚೆ ಅಲ್ಲಿ ಮರಗಳು ಹಸಿರಾಗಿದ್ವು, ಮತ್ತೆ ಮರಗಳ ಮೇಲೆ ಆಲ್ಗೆ ಬೆಳ್ಕೊಂಡಿತ್ತು. ಅಲ್ಲಿ ಇರುವಂತ ಕೆಲವು ಜೀವಿಗಳ ಬಗ್ಗೆ ಹೇಳ್ತಾ ಇದ್ದಾರೆ. ಸೊ ದಿಸ್ ಇಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಪೆಪ್ಪರ್ಡ್ ಮಾತ್ ಅಂತ ಕರೀತಾರೆ. ಈ ಕೀಟಗಳನ್ನ ಪೆಪ್ಪರ್ಡ್ ಮಾತ್ ಅಂತ ಕರೀತಾರೆ. ಇಲ್ಲ ನೋಡಿ, ಈ ಕಡೆ ಪಿಕ್ಚರ್ ಅಲ್ಲಿ ವೈಟ್ ಪೆಪ್ಪರ್ಡ್ ಮಾತಿನ ಸಂಖ್ಯೆ ಜಾಸ್ತಿ ಇದೆ. ಅಂದ್ರೆ ಏನರ್ಥ, ಇಂಡಸ್ಟ್ರಿಯಲೈಸೇಷನ್ ಆಗಕ್ಕಿಂತ ಮುಂಚೆ ಆ ಹೊಗೆ ಇರ್ತಿರ್ಲಿಲ್ಲ, ಪೊಲ್ಯೂಷನ್ ಇರ್ತಿರ್ಲಿಲ್ಲ, ಯಾವ ತರ ಒಂದು ಎಫೆಕ್ಟ್ ಇರದೇ ಇರೋ ಕಾರಣ ಮರಗಳಲ್ಲಿ ಲೈಕನ್ಸ್ ಚೆನ್ನಾಗಿ ಬೆಳ್ಕೊಂತ ಇತ್ತು. ದ ಲೈಕನ್ಸ್ ವರ್ ಗ್ರೋಯಿಂಗ್ they were flourishing very good on the trees. So when these insects would go and sit on this, the birds could not identify the white moths, but there was another strain, ಡಾರ್ಕ್ ಪೆಪ್ಪರ್ಡ್ ಮಾತ್ಗಳಿದ್ವು. ಸೊ ಏನಾಗ್ತಿತ್ತು, ಇವೆರಡೂ ಮರದ ಮೇಲೆ ಕೂತ್ಕೊಂಡಾಗ ಇದು ಕಾಣಿಸ್ತಾ ಇರ್ಲಿಲ್ಲ ಪಕ್ಷಿಗಳಿಗೆ, ಆದ್ರೆ ಇದು ನೀಟಾಗಿ ಕಾಣಿಸ್ತಾ ಇತ್ತು. ಸೊ ಪಕ್ಷಿಗಳು ಯಾರನ್ನ ಆರಿಸ್ಕೊಂತಾ ಇದ್ರು, ಇದನ್ನ ಆರಿಸ್ಕೊಂಡು ಡಾರ್ಕ್ ಕಲರ್ಡ್ ಪೆಪ್ಪರ್ಡ್ ಮಾತ್ ನ ತಿಂದು ಹಾಕ್ತಾ ಇದ್ರು. ಹಾಗಾಗಿ ಇದರ ಪಾಪ್ಯುಲೇಷನ್ ಜಾಸ್ತಿ ಇತ್ತು. ಓಕೆ, ಸೊ ಇಲ್ಲಿ ನೋಡಿ, ಇಲ್ಲಿ ಬರ್ದಿದೀವಿ 10% ಕಲರ್ಡ್ ಡಾರ್ಕ್ ಕಲರ್ಡ್ ಫಿನೋಟೈಪ್. ಫಿನೋಟೈಪ್ ಅಂದ್ರೆ ಒಂದು ಕ್ಯಾರೆಕ್ಟರ್. ಸೊ 10% ಅಷ್ಟೇ ಅವ್ರ ಪಾಪ್ಯುಲೇಷನ್ ಇದೆ. 90% ಫುಲ್ ವೈಟ್ ಕಲರ್ಡ್ ಪೆಪ್ಪರ್ಡ್ ಮಾತಾ ಇದ್ರು. ಬಟ್ ಮುಂದಕ್ಕೆ ಏನಾಯ್ತು, ಇಂಡಸ್ಟ್ರಿಯಲೈಸೇಷನ್ ಆದ್ಮೇಲೆ ಏನಾಯ್ತು, ಇಂಡಸ್ಟ್ರಿಯಲೈಸೇಷನ್ ಆದ್ಮೇಲೆ ಈ ಮರದ ಮೇಲೆ ಲೈಕನ್ಸ್ ಎಲ್ಲ ಮಾಯ ಆಗೋಗಿದೆ. ನೋಡಿ ಕಾಣಿಸ್ತಾ ಇದೆ, ಬ್ಲಾಕ್ ಮರ ಆಗ್ಬಿಟ್ಟಿದೆ, ಫುಲ್ ಫಾಗ್ ತುಂಬ್ಕೊಂಡ್ಬಿಟ್ಟಿದೆ, ಸೂಚ್ ತುಂಬ್ಕೊಂಡು ಏನೋ ಬರೇ ಹೊಗೆ ತುಂಬ್ಕೊಂಡ್ಬಿಟ್ಟಿದೆ ಮರದ ತುಂಬಾ. ಸೊ ಲೈಕನ್ಸ್ ಎಲ್ಲ ಸತ್ತೋಗಿದೆ. ಇದ್ರ ಮೇಲೆ ಈ ಮಾತ್ಗಳು ಕೂತಾಗ ಪಕ್ಷಿಗಳು ಯಾರನ್ನ ಸೆಲೆಕ್ಟ್ ಮಾಡ್ತಾರೆ, ವೈಟ್ ಪೆಪ್ಪರ್ಡ್ ಮಾತ್ ನ ಸೆಲೆಕ್ಟ್ ಮಾಡಿದೆ, ಯಾಕಂದ್ರೆ ನಿಮ್ಗೆ ಕಾಣಿಸೋದಿಲ್ಲ. ಸೊ ಅವಾಗ ಇದರ ಪಾಪ್ಯುಲೇಷನ್ ಜಾಸ್ತಿ ಆಯ್ತು. ಯಾವ್ದ್ರದ್ದು, ಡಾರ್ಕ್ ಕಲರ್ಡ್ ಪೆಪ್ಪರ್ಡ್ ಮಾತ್ ಪಾಪ್ಯುಲೇಷನ್ ಜಾಸ್ತಿ ಆಯ್ತು. ವೈಟ್ ಕಲರ್ಡ್ ಪೆಪ್ಪರ್ಡ್ ಮಾತ್ ಪಾಪ್ಯುಲೇಷನ್ ಪೂರಾನೇ ಕಡಿಮೆ ಆಯ್ತು. ಸೊ ನೇಚರ್ಗೆ ಯಾರು ಅಡಾಪ್ಟ್ ಆಗ್ತಾರೋ, ನೇಚರ್ ಜೊತೆ ಯಾರು ಸ್ಪಂದಿಸ್ತಾರೋ ಅವ್ರನ್ನ ಮಾತ್ರ ನೇಚರ್ ಸೆಲೆಕ್ಟ್ ಮಾಡುತ್ತೆ. ಅದನ್ನೇ ನ್ಯಾಚುರಲ್ ಸೆಲೆಕ್ಷನ್ ಅಂತ ಕರೀತಾರೆ. ಇದು ತುಂಬಾ ಇಂಪಾರ್ಟೆಂಟ್ ಮಕ್ಕಳ. ಎಕ್ಸಾಮ್ಗೆ 5 ಮಾರ್ಕ್ಸ್ ಕೇಳ್ತಾರೆ ಈ ಟಾಪಿಕ್ ಇಂದ. ಯಾವ ತರ ಕೇಳ್ತಾರೆ ಅಂದ್ರೆ, ಎಕ್ಸ್ಪ್ಲೈನ್ ನ್ಯಾಚುರಲ್ ಸೆಲೆಕ್ಷನ್ ವಿತ್ ದ explain natural selection with an example of industrial melanism. ಮೆಲಾನಿಸಮ್ ಅಂದ್ರೆ ಮೆಲಾನಿನ್ ಪಿಗ್ಮೆಂಟ್ ಹೇಗೆ ಡಿಸ್ಟ್ರಿಬ್ಯೂಟ್ ಆಗಿದೆ ನೋಡಿ. ಇಲ್ಲಿ ಮೆಲಾನಿನ್ ಪಿಗ್ಮೆಂಟ್ ಇಲ್ಲಿ ಇಲ್ವೇ ಇಲ್ಲ, ಕಡಿಮೆ ಇದೆ, ವೈಟ್ ಆಗಿದೆ. ಅದಕ್ಕೆ ಇಲ್ಲಿ ಜಾಸ್ತಿ ಮೆಲಾನಿನ್ ಪಿಗ್ಮೆಂಟ್ ಇರೋದ್ರಿಂದ ಇದು ಡಾರ್ಕ್ ಆಗಿದೆ. ಸೊ ಇದು ಗೆಲ್ತು. ಇದು ನೇಚರ್ ನ ಇದು ಸೆಲೆಕ್ಟ್ ಮಾಡಿ, ನೇಚರ್ ಇದನ್ನ ಸೆಲೆಕ್ಟ್ ಮಾಡಿದೆ, ಡಾರ್ಕ್ ಕಲರ್ ಪೆಪ್ಪರ್ಡ್ ಮಾತ್ ನ ಸೆಲೆಕ್ಟ್ ಮಾಡ್ಕೊಂಡಿದೆ, ವೈಟ್ ಕಲರ್ ಪೆಪ್ಪರ್ಡ್ ಮಾತ್ ನ ರಿಜೆಕ್ಟ್ ಮಾಡಿದೆ. ಸೊ ದಟ್ ಇಸ್ ಅ ವೆರಿ ಗುಡ್ ಎಕ್ಸಾಂಪಲ್ ಟು ಸೇ ಹೌ ದ ನೇಚರ್ ಸೆಲೆಕ್ಟ್ಸ್ the individuals. So let us move on to next our story. What is our story? Evolution of human. So usually ನಾವು ಮಕ್ಕಳೆಲ್ಲಾರ ತರಲೆ ಮಾಡ್ತಿದ್ರೆ ಏನ್ ಹೇಳ್ತೀವಿ, ನಿನ್ ಮಂಗನ್ ತರ ಆಡ್ತೀಯ ಅಂತ. So that is because we all have that character in us. ನಾವೆಲ್ಲರಲ್ಲೂ ಕೂಡ ಒಂದು ಚಿಕ್ಕ ಮಂಗ ಕೂತಿದೆ. ಯಾಕಂದ್ರೆ we all come from apes. We are all arrived on this earth ಫ್ರಮ್ ಏಪ್ಸ್. ಅಂದ್ರೆ ನಾವೆಲ್ಲ ಹುಟ್ಟಿರೋದು ನಮ್ಮ ಆನ್ಸಿಸ್ಟರ್ ಏಪ್ಸ್ ಗಳಿಂದ, ಮಂಗಗಳಿಂದನೇ ನಾವು ಬಂದಿರೋದು. ಇದು ಹೇಗೆ ಗೊತ್ತಾಯ್ತು? ಫಾಸಿಲ್ಸ್ ಇಂದ. ಸೊ ವಿ ಹ್ಯಾವ್ ಕಲೆಕ್ಟೆಡ್ ಸೋ ಮೆನಿ ಫಾಸಿಲ್ಸ್ ಟು ಸೇ ದಟ್ ವಿ ಹ್ಯಾವ್ ಎವಾಲ್ವ್ಡ್ ಫ್ರಮ್ ಏಪ್ಸ್. ಸೊ ಲೆಟ್ ಅಸ್ ಸ್ಟಡಿ ಹೌ ವಿ ಹ್ಯಾವ್ ಎವಾಲ್ವ್ಡ್ ಫ್ರಮ್ ಏಪ್ಸ್. ಸೊ ಹಿಯರ್ ದಿಸ್ ಇಸ್ ದ ಫಸ್ಟ್ ಸ್ಟೇಜ್ ರೆಕಾರ್ಡೆಡ್. ಅಂದ್ರೆ ಇದಕ್ಕಿಂತ ಮುಂಚೆ ತುಂಬಾ ಸ್ಟೇಜ್ಗಳಿದೆ, ಬಟ್ ರೆಕಾರ್ಡೆಡ್ ಸ್ಟೇಜ್ ಯಾವುದು ಅಂದ್ರೆ ಡ್ರಯೋಪೆಥಿಕಸ್. ಸೊ ಡ್ರಯೋಪೆಥಿಕಸ್ ಅನ್ನೋ ಒಂದು ಜೀವಿ ಅದು ಏಪ್ ತರನೇ ಇತ್ತು. ಸೊ ಅದು ಏಪ್ ತರನೇ ಇರುವಂತ ಒಂದು ಜೀವಿ. ಸೊ ಇದು ಸುಮಾರು ಡ್ರಯೋಪೆಥಿಕಸ್ ಅನ್ನೋ ಒಂದು ಜೀವಿ ಸುಮಾರು 25 ಮಿಲಿಯನ್ ಇಯರ್ಸ್ ಎಗೋ ಭೂಮಿ ಮೇಲಿತ್ತು. ಸೊ ಇದು ನೋಡೋದಕ್ಕೆ ಸೇಮ್ ಮಂಕಿ ತರನೇ ಇತ್ತು. ಸೇಮ್ ಅಂದ್ರೆ 4 ಕಾಲುಗಳಲ್ಲಿ ಓಡಾಡ್ತಾ ಇತ್ತು. ಇಟ್ ವಾಸ್ ನಾಟ್ ಲೈಕ್ ಹ್ಯೂಮನ್ ವಿತೌಟ್ ಎರೆಕ್ಟ್ ಪಾಸ್ಚರ್. ಮತ್ತೆ ಇದರ ಕೆನೈನ್ಸ್ ತುಂಬಾ ಅಗಲವಾಗಿತ್ತು, ಯಾಕೆಂದ್ರೆ ದೇ ವರ್ ಫೀಡಿಂಗ್ ಆನ್ ದ ಮೀಟ್. ಆಮೇಲೆ they didn't have any behavior, nothing. So they were very much similar to apes. ಇವರಿಂದ ನೆಕ್ಸ್ಟ್ ಹುಟ್ಕೊಂಡಿದ್ದು ಯಾರು? 14 to 15 ಮಿಲಿಯನ್ ಇಯರ್ಸ್ ಎಗೋ ರಾಮಾಪೆಥಿಕಸ್. ಯಾಕೆ ರಾಮಾಪೆಥಿಕಸ್ ಅಂದ್ರೆ ಇದರ ಫಾಸಿಲ್ಸ್ ನಮ್ಮ ದೇಶ ಅಂದ್ರೆ ಭಾರತ ದೇಶದ ಶಿವಲಿಂಗ್ ಅನ್ನೋ ಒಂದು ಬೆಟ್ಟಗಳ ಮಧ್ಯದಲ್ಲಿ ಸಿಕ್ಕಿರೋದ್ರಿಂದ ಇದನ್ನ ರಾಮಾಪೆಥಿಕಸ್ ಅಂತ ಕರೀತಾರೆ. ಸೊ ಇಟ್ ಇಸ್ ನೋವೇ ರಿಲೇಟೆಡ್ ಟು ರಾಮ. ರಾಮನ್ಗೂ ಇದಕ್ಕೂ ಸಂಬಂಧನೇ ಇಲ್ಲ. ಇಂಡಿಯಾನಲ್ಲಿ ಸಿಕ್ಕಿರೋದ್ರಿಂದ ರಾಮಾಪೆಥಿಕಸ್ ಅಂತ ಕರೀತಾರೆ. ಸೊ ದಿಸ್ ಅನಿಮಲ್ ವಾಸ್ ಹ್ಯಾವಿಂಗ್ ರೆಡ್ಯೂಸ್ಡ್ ಕೆನೈನ್ಸ್ ಚಿಕ್ಕದಾಗಿತ್ತು. ಆಮೇಲೆ ಮೋಲಾರ್ಸ್ ಎಲ್ಲ ಫ್ಲಾಟ್ ಆಗಿತ್ತು. ಸೊ ದೇ ವರ್ ಲಿಟಲ್ ಲಿಟಲ್ ಓಮ್ನಿವರಸ್. ಅಂದ್ರೆ ಏನೋ ಮೀಟ್ ಬಿಟ್ಟು ಸೀಡ್ಸ್, ನಟ್ಸ್, ಫ್ರೂಟ್ಸ್ ನ ತಿನ್ನೋಕೆ ಸ್ಟಾರ್ಟ್ ಮಾಡಿದ್ರು. ದ ನೆಕ್ಸ್ಟ್ ಒನ್ ಈಸ್ ಆಸ್ಟ್ರಲೋಪೆಥಿಕಸ್. ಈ ಆಸ್ಟ್ರಲೋಪೆಥಿಕಸ್ ದೇ ಲಿವ್ಡ್ ಸಮ್ವೇರ್ ಅರೌಂಡ್ 5 ಮಿಲಿಯನ್ ಇಯರ್ಸ್ ಎಗೋ. ಸೊ ಈ ಆಸ್ಟ್ರಲೋಪೆಥಿಕಸ್ ನ ಏಪ್ ಮ್ಯಾನ್ ಅಂತ ಕರೀತಾರೆ. ಏಪ್ ಮ್ಯಾನ್ ಅಂದ್ರೆ ಸ್ವಲ್ಪ ಏಪ್ ತರನೂ ಇತ್ತು, ಸ್ವಲ್ಪ ಮನುಷ್ಯನ್ ತರನೂ ಇತ್ತು. ಅಂಡ್ ದೇ ವರ್ ವೆರಿ ಮಚ್ ಸಿಮಿಲರ್ ಟು ದಿಸ್ ಮ್ಯಾನ್ ಹೂ ಇಸ್ ಸ್ಟ್ಯಾಂಡಿಂಗ್ ಹಿಯರ್. ಎರೆಕ್ಟ್ ಪಾಸ್ಚರ್ ಇತ್ತು. ಅವ್ರಿಗೆ ಸ್ಟ್ರೈಟ್ ಆಗಿ ನಿಲ್ತಾ ಇದ್ರು. ಎರಡೇ ಕಾಲಲ್ಲಿ ನಡೆಯುವಂತ ಒಂದು ಶಕ್ತಿ ಅವ್ರಿಗಿತ್ತು. ಸೊ ದೀಸ್ ದೇ ಹ್ಯಾಡ್ ಅ ವೆರಿ ಎರೆಕ್ಟ್ ಪಾಸ್ಚರ್. ಅಂಡ್ ನೆಕ್ಸ್ಟ್ ವಾಸ್ ಹೋಮೋ ಹ್ಯಾಬಿಲಸ್. ಹೋಮೋ ಹ್ಯಾಬಿಲಸ್ ಯಾಕೆ ಅಂದ್ರೆ ದೇ ವರ್ ಲಿವಿಂಗ್ ಇನ್ ಹೌಸ್, ತಮ್ಮದೇ ಆದ ಒಂದು ಮನೆಗಳು ಮಾಡ್ಕೋತ, ಮನೆ ಅಂದ್ರೆ ಕೇವ್ಸ್, ಗುಹೆ ಒಳಗಡೆ ವಾಸ ಮಾಡ್ತಾ ಇದ್ರು. ಆಮೇಲೆ ತಮಗ್ ಸಿಕ್ತಿದ್ದಂತ ಕಲ್ಲುಗಳಿಂದ ಟೂಲ್ಸ್ ಗಳು ಮಾಡ್ತಾ ಇದ್ರು, ಅದರಿಂದ ಹಂಟ್ ಮಾಡ್ತಾ ಇದ್ರು, ಅದರಿಂದ ಚರ್ಮಗಳನ್ನ ತಗೊಂತಾ ಇದ್ರು. ಸೊ ದಟ್ ವಾಸ್ ದೇರ್ ಹ್ಯಾಬಿಟ್. ಸೊ ಅದಕ್ಕೆ ಹೋಮೋ ಹ್ಯಾಬಿಲಸ್ ನ ಟೂಲ್ ಮೇಕರ್ ಅಂತಾನು ಕರೀತಾರೆ. ಟೂಲ್ಸ್ ಮಾಡ್ತಾ ಇದ್ರು ಸ್ಟೋನ್ಸ್ ಗಳಿಂದ, ಬೋನ್ಸ್ ಗಳಿಂದ, ಅವ್ರು ಸಾಯಿಸಿರುವಂತ ಒಂದು ಪ್ರಾಣಿಗಳಿಂದ ಟೂಲ್ಸ್ ಗಳನ್ನ ಮಾಡ್ತಾ ಇದ್ರು. ಸೊ ಅದಕ್ಕೆ ಅವ್ರನ್ನ ಹೋಮೋ ಹ್ಯಾಬಿಲಸ್ ಅಂತ ಕರೀತಾರೆ. ಸೊ ದೇ ವರ್ ಶೋಯಿಂಗ್ ಸಮ್ ಸೋಷಿಯಲ್ ಬಿಹೇವಿಯರ್. ಸಮ್ ಸೋಷಿಯಲ್ ಬಿಹೇವಿಯರ್ ಅಂದ್ರೆ ಸೋಷಿಯಲ್ ಬಿಹೇವಿಯರ್ ಇತ್ತು, ಗುಂಪಲ್ಲಿ ಇರೋದು, ಆ ತರ ಒಬ್ಬರನ್ನೊಬ್ರು ಪ್ರೊಟೆಕ್ಟ್ ಮಾಡ್ಕೊಂಡಿರೋದು, ಯಾರ ಬಂದು ಅವ್ರನ್ನ ಅಟ್ಯಾಕ್ ಮಾಡಿದ್ರೆ ಅವ್ರನ್ನ ದೂರ ಹಾಕೋದು, ಆ ತರ ಗುಂಪು, ಆ ತರ ಸೋಷಿಯಲ್ ಬಿಹೇವಿಯರ್ ನ ನಾವು ಇವರಲ್ಲಿ ಗಮನಿಸಬಹುದಾಗಿತ್ತು. ನೆಕ್ಸ್ಟ್ ಯಾವುದು? ಹೋಮೋ ಎರೆಕ್ಟಸ್. ಆಸ್ ದ ನೇಮ್ ಸೈಸ್ ಎರೆಕ್ಟ್. ಸೊ ಎರೆಕ್ಟ್ ಪಾಸ್ಚರ್ ಇತ್ತು, ಅವರ ಸ್ಪೈನ್ ಅಲ್ಲಿ ಕರ್ವ್ ಇತ್ತು. ದೇ ವರ್ ವೆರಿ ಮಚ್ ಸಿಮಿಲರ್ ಟು ಹ್ಯೂಮನ್ಸ್. ಹೋಮೋ ಎರೆಕ್ಟಸ್ ಇವ್ರನ್ನ ಜಾವಾ ಮ್ಯಾನ್ ಅಂತಾನು ಕರೀತಾರೆ. ಯಾಕೆಂದ್ರೆ ಜಾವಾ ಅನ್ನೋ ಒಂದು ಜಾಗದಲ್ಲಿ ಇವರ ಸ್ಕೆಲಿಟನ್ ಸಿಕ್ಕಿದೆ, ಸ್ಕಲ್ ಸಿಕ್ಕಿದೆ, ಅದಕ್ಕೆ ಅವ್ರನ್ನ ಜಾವಾ ಮ್ಯಾನ್ ಅಂತೂ ಕರೀತಾರೆ, ಮತ್ತೆ ಹೋಮೋ ಎರೆಕ್ಟಸ್ ಅಂತೂ ಕರೀತಾರೆ ಮಕ್ಳ. ಸೊ ದೇ ವರ್ ನೋನ್ ಫಾರ್ ಹಂಟಿಂಗ್. ಇವರು ಅಷ್ಟೇ, ಬೋತ್ ವರ್ ವೆರಿ ಗುಡ್ ಇನ್ ಹಂಟಿಂಗ್. ದೇ ವರ್ ಯೂಸಿಂಗ್ ದ ಸ್ಟೋನ್ಸ್ for hunting the organisms. They were making spades, they were making arrows and all that from the bones, and they were hunting the organisms. That is about the Homo erectus. And these Homo erectus, they were very very similar to human. Tumbane similarity ittu. They lived for about 1.7 million years ago. These were the animals which were seen, Homo erectus. ನೆಕ್ಸ್ಟ್ ಅದಾದ್ಮೇಲೆ ಬಂದಿದ್ದೆ ಹೋಮೋ ಸೆಪಿಯನ್ಸ್. ಇದು ಮಧ್ಯದೇನ್ ಬೇಕಾಗಿಲ್ಲ. ಹೋಮೋ ಸೆಪಿಯನ್ಸ್ ಹ್ಯಾವ್ ಅರೈವ್ಡ್ ಆನ್ ಅರ್ತ್ somewhere around 25,000 years ago. They are the modern man. Modern man ಯಾಕೆಂದ್ರೆ so they were able to have culture, they had their own culture, they were able to speak to each other by some languages. They were also very good in cultivating the lands, agriculture. Okay, they were hunting, they were domesticating the animals. So that is how they came up. And ಇವಾಗ್ಲೂ ಕೂಡ ಈ ಹೋಮೋ ಎರೆಕ್ಟಸ್ ಇದೆಯಲ್ಲ, ಈ ಹೋಮೋ ಎರೆಕ್ಟಸ್ ಗಳು ಕೇವ್ಸ್ ಗಳಲ್ಲಿ ಪೇಂಟ್ಸ್ ನ ಮಾಡ್ತಾ ಇದ್ರು. ಅಂದ್ರೆ ಪ್ರಾಣಿಗಳನ್ನ ಅವ್ರು ಸಾಯಿಸ್ತಾರಲ್ಲ, ಅದ್ರ ಪೇಂಟ್ ಇಂದ ಅವರು ಕಾರ್ವಿಂಗ್ಸ್ ಮಾಡ್ತಾ ಇದ್ರು. ನೀವು ಅಜಂತಾ ಎಲ್ಲೋರಾಗ ಇವಾಗ್ಲೂ ಹೋದ್ರೆ ಮಕ್ಳ, ಅಲ್ಲಿ ಪೇಂಟ್ ಕಾರ್ವ್ ಕಾಣಿಸುತ್ತೆ. ಅಂದ್ರೆ ಆಗಿನ ಕಾಲದ ಜನಗಳು ಪೇಂಟ್ ಮಾಡಿರೋ ಕಾರ್ವಿಂಗ್ಸ್ ನ ನೀವು ನೋಡ್ಬೋದು. ಸೊ ಅದನ್ನ ಪೇಂಟಿಂಗ್ಸ್ ಅಂತ ಕರೀತಾರೆ. ಆಗಿನ ಕಾಲದ ಜನಗಳು ಪೇಂಟಿಂಗ್ ಮಾಡಂದ್ರೆ ಅವ್ರು ಏನ್ ಅವತ್ ಮಾಡಿದ್ರು, ಯಾವ ತರ ಅನಿಮಲ್ ಸಾಯಿಸಿದ್ರೆ, ಅದರ ಪ್ರಾಣಿನ ಸಾಯಿಸಿದ್ರೆ ಅದನ್ನ ಅವ್ರು ಬರೀತಾ ಇದ್ರು. ಸೊ ದಟ್ ಈಸ್ ವೆರಿ ಫೇಮಸ್ ಅಬೌಟ್ ಕೇವ್ ಪೇಂಟಿಂಗ್ಸ್ ಅಂತ ಕರೀತಾರೆ ಅಜಂತಾ ಎಲ್ಲೋರಲ್ಲಿ. ಸೊ ದಟ್ ಇಸ್ ವೆರಿ ಇಂಪಾರ್ಟೆಂಟ್ ಥಿಂಗ್ ಅಬೌಟ್ ಹೋಮೋ ಎರೆಕ್ಟಸ್. So this is also another picture which shows about evolution of man. So you can see how man has got evolved. So this is Australopithecus, Homo habilis. They were able to speak. They were able to speak by some of their languages. Ivaglu tribals ko adra nevenno speech noಡ್ತಿರಲ್ಲ, and these were hunting. Homo erectus, see, they are very much similar to human, but you can see the ridges are very prominent, and you can see the chin is not very prominent still. But alla nodta iri facial features na nodta hogi makla, you can see lot of difference in the facial features. They hunted in groups. Homo erectus ಅವರು they hunted in groups ಆಗಿ hunt ಮಾಡ್ತಿದ್ರು. They started living in communities. ಒಂದೊಂದು ಗುಂಪುಗಳಾಗಿ ವಾಸ ಮಾಡಕ್ಕೆ ಸ್ಟಾರ್ಟ್ ಮಾಡಿದ್ವು. Then you have Neanderthal man ಅಥವಾ ಹೋಮೋ ಏನಂತಾರೆ, this is the Neanderthal man. ಇದು they are called as the first civilized man. First civilized man ಯಾಕೆ ಅಂದ್ರೆ they were burying the bodies. ಅಂದ್ರೆ ಯಾರ ಸತ್ತೋದ್ರೆ, ಯಾರ ತೀರೋದ್ರೆ ಆ ಬಾಡಿಗೆ ರಿಚುವಲ್ಸ್ ಮಾಡಿ, ಅಂದ್ರೆ ಏನೇನು ಮಾಡ್ಬೇಕು ಅದೆಲ್ಲ, ಅಂದ್ರೆ ರಿಚುವಲ್ಸ್ ಅಂದ್ರೆ ಏನೋ, ಅದಕ್ಕೆ ಮಂತ್ರ ಹಾಕಿ ಅಥವಾ ಏನೋ ಮಾಡಿ ಅದನ್ನ ಬರಿ ಮಾಡ್ತಾ ಇದ್ರು. ತುಂಬಾ ಪ್ರೈಡ್ ಇಂದ ಬಾಡಿಗಳನ್ನ ಬರಿ ಮಾಡ್ತಾ ಇದ್ರು. ದೇ ವರ್ ಯೂಸಿಂಗ್ ಟೂಲ್ಸ್. ಕಾಣಿಸ್ತಿದ್ಯಾ, ಅವರ ಕೈಯಲ್ಲಿ ಒಂದು ಟೂಲ್ ಇದೆ ಕಾಣಿಸ್ತಾ, ದಿಸ್ ಇಸ್ ಮೇಡ್ ಅಪ್ ಆಫ್ ಬೋನ್. ಸೊ ಆ ತರ ದೇ ವರ್ ಶೋಯಿಂಗ್ ಕಲ್ಚರ್ ಅನ್ನೋದನ್ನ ನೋಡ್ತಾ ಇದ್ವಿ. ಕಮಿಂಗ್ ಆನ್ ಟು ಹೋಮೋ ಸೆಪಿಯನ್ಸ್, ದೇ ಆರ್ ದ ವೆರಿ ವೆರಿ ಹತ್ರದ ಆನ್ಸಿಸ್ಟರ್ ಅಂತ ಹೇಳ್ಬೋದು, ತುಂಬಾ ಹತ್ರದ ರಿಲೇಟಿವ್ ಅವರು ಹೋಮೋ ಸೆಪಿಯನ್ಸ್. ಯಾಕೆಂದ್ರೆ ದೇ ವರ್ ವೆರಿ ಗುಡ್ ಇನ್ ಆರ್ಟ್, tool making, and they were good for cultivating the plants. They were good in domesticating the animals. So they were very near to what the modern man is. So that is about the evolution of man. So tumba important makla. ಹೇಗೆ ಇಂಪಾರ್ಟೆಂಟ್ ಅಂದ್ರೆ ಆರ್ಡರ್ ಗೊತ್ತಿರ್ಬೇಕು. ಫಸ್ಟ್ ಬರೋದು ಡ್ರಯೋಪೆಥಿಕಸ್, ರಾಮಾಪೆಥಿಕಸ್, ಆಸ್ಟ್ರೇಲೋಪೆಥಿಕಸ್, ಹೋಮೋ ಹ್ಯಾಬಿಲಸ್, ಹೋಮೋ ಎರೆಕ್ಟಸ್, ಆಮೇಲೆ ನೆಕ್ಸ್ಟ್ ಲಾಸ್ಟ್ ಬರೋದು ಹೋಮೋ ಸೆಪಿಯನ್ಸ್. ಓಕೆ, ಸೊ ಇದ್ರ ಮಧ್ಯೆ ಒಂದಿದೆ ಹೋಮೋ ಸೆಪಿಯನ್ಸ್ ಅಂತ ಇದೆಯಲ್ಲ, ಅಲ್ಲಿ ನಿಯೋನ್ಯಾಂಡ್ರೆಥಲ್ ಮ್ಯಾನ್, ಇಲ್ಲೊಂದಿದೆ ನೋಡಿ ನಿಯೋನ್ಯಾಂಡ್ರೆಥಲ್ ಮ್ಯಾನ್ ಅನ್ನೋದನ್ನ ಆಡ್ ಮಾಡ್ಕೋಬೇಕು. ಸೊ ಡ್ರಯೋಪೆಥಿಕಸ್, ರಾಮಾಪೆಥಿಕಸ್, ಆಸ್ಟ್ರೇಲೋಪೆಥಿಕಸ್, ಹೋಮೋ ಹ್ಯಾಬಿಲಸ್, ಹೋಮೋ ಎರೆಕ್ಟಸ್ ಅಥವಾ ನಿಯೋಆಂಡ್ರೆಥಲ್ ಮ್ಯಾನ್ ಅಂತಾರೆ. ನೆಕ್ಸ್ಟ್ ಹೋಮೋ ಸೆಪಿಯನ್ಸ್. ಸೊ ಈ ಆರ್ಡರ್ ನ ಎಕ್ಸಾಮ್ ಅಲ್ಲಿ MCQ ನಲ್ಲಿ ಕೇಳಬಹುದು. ಸೊ ಅದನ್ನ ಜ್ಞಾಪಕ ಇಟ್ಕೊಳ್ಳಿ. DRAHH ಆ ತರ ಒಂದು ಏನಾರ ಮಾಡ್ಕೊಂಡು ಅದನ್ನ ಆರ್ಡರ್ ನ ಜ್ಞಾಪಕ ಇಟ್ಕೊಳ್ಳಿ. ಸೊ ದಿಸ್ ಇಸ್ ಅ ವೆರಿ ಇಂಪಾರ್ಟೆಂಟ್ ಕ್ವೆಶ್ಚನ್ ಫ್ರಮ್ ಎವಲ್ಯೂಷನ್ ಚಾಪ್ಟರ್. Okay, so that is about the evolution of man. So this is the evolutionary tree. So you can see how the evolution has occurred. So first we had the bacteria's kingdom Monera. Then from kingdom Monera we got kingdom Protista. All the organisms have come from kingdom. You can see the coelentrates, the dinoflagellates and all. Then kingdom Mycota. So last session was about kingdom Mycota. So you can see all the fungi coming into existence. Then you have the angiosperms, the gymnosperms. Then you have the animals coming into existence. First the invertebrates, and there we stand. So we vertebrates are at the end of the existence. So that is where we stand in this evolutionary tree. So we should know that we have come from the base root. We have come from the basic root of our existence. So we have come from a very, very primitive state of cell. And now we have got complicated to become a vertebrate or a human. So this is the evolutionary tree which shows how the life existed, what all the organisms are there on this earth, in a very simple manner. So that is about, and always I have a question. That question in me is, evolution of man, is it good or bad? So always I have this question in my mind, should we had evolved? Now evolve ಆಗ್ಬೇಕಾಗಿತ್ತಾ evolution? Because we can see a lot of things are happening because of human evolution. We are cutting the trees, we are flooding the earth, we are polluting the earth. Not only earth, we are polluting the space also. We are destroying the entire earth. So that is why the question always remains in my mind, is evolution of man good or bad? So that is about my seminar. Thank you very much. Sir?
Thank you, madam.
Thank you, sir.
ವಿದ್ಯಾರ್ಥಿಗಳೇ,
ಕ್ವಶನ್ಸ್ ಇದ್ರೆ ಕೇಳಬಹುದು ಮಕ್ಕಳು.
Dear students, please ask any questions about today's topic. ಯಾವುದೇ ಡೌಟ್ಸ್ ಅಥವಾ ಕ್ವಶನ್ಸ್ ಗಳಿದ್ರೆ ಚಾಟ್ ಬಾಕ್ಸ್ ಅಲ್ಲಿ ಒಂದ್ಸರಿ ಚೆಕ್ ಮಾಡಿ. ಮೇಡಮ್, ಮಕ್ಕಳು ಯಾವುದೇ ಕ್ವಶನ್ಸ್ ಚಾಟ್ ಬಾಕ್ಸ್ ಅಲ್ಲಿ ಹಾಕಿಲ್ಲ.
ಓಕೆ ಸರ್, ಆಯ್ತು. ಓಕೆ,
ಸೊ ಲೇಟರ್
ಕ್ವಶನ್ಸ್ ಹಾಕಿದ್ರೆ
I
ಕೇಳ್ತೀನಿ.
sent through mail, ma'am.
ಓಕೆ ಸರ್, ಥ್ಯಾಂಕ್ ಯು ಸರ್.
ಮೇಲ್ ಮುಖಾಂತರ ಕಳಿಸ್ಕೊಡ್ತೀನಿ.
Yes, sir. I want to thank KSTA. I want to thank Ramesh sir and Ayappan sir and Siddegowda sir for giving me an opportunity to talk about evolution. And I thank a lot for KSTA to give me that courage to speak online. Thanks a lot, KSTA.
Thank you a lot, ma'am.
Thank you a lot, sir.
ಸೊ ತಾವು ಕೊಟ್ಟಂತ ಈ ಒಂದು ಉಪನ್ಯಾಸ ಕಾರ್ಯಕ್ರಮವು ಮಾನವನ ವಿಕಾಸ ಯಾವ ರೀತಿ ಆಯಿತು, ಅದು ಕೊನೆಗೆ ಯಾವ ರೀತಿ ಬಂದು ನಿಂತಿದೆ ಎನ್ನುವ ಒಂದು ಉಪನ್ಯಾಸ ಕಾರ್ಯಕ್ರಮವನ್ನು ತಾವು ವಿದ್ಯಾರ್ಥಿಗಳಿಗೆ ಸೊ ಡೆಲಿವರ್ಡ್ ಎ ವೆರಿ ಗುಡ್ ಇನ್ಫಾರ್ಮೇಟಿವ್ ಟಾಕ್ ಟು ಆಲ್ ದಿ ಫಸ್ಟ್ PUC and second PUC ಸ್ಟೂಡೆಂಟ್ಸ್, ಮ್ಯಾಮ್. ಸೊ ಹಾರ್ಟ್ಲಿ ನನ್ನ ವೈಯಕ್ತಿಕವಾಗಿ ಹಾಗೂ ಮತ್ತು ನನ್ನ KSTA ಪರವಾಗಿ ಧನ್ಯವಾದಗಳು ಮೇಡಮ್.
ಥ್ಯಾಂಕ್ ಯು ಸರ್. ಥ್ಯಾಂಕ್ ಯು KSTA ಸರ್. ಸೊ
ಹಾಗೂ ನಮ್ಮ ಅಕಾಡೆಮಿಯ ಅಧ್ಯಕ್ಷರಾದಂತ ಪ್ರೊಫೆಸರ್ ಅಯ್ಯಪ್ಪನ್ ಸರ್ಗೂ ಕೂಡ ಧನ್ಯವಾದಗಳು ಸರ್. ಹಾಗೂ ಈ ಒಂದು ವಿಜ್ಞಾನ ಪ್ರತಿಭಾ ಶೋಧನೆ ಯೋಜನೆ ಕಾರ್ಯಕ್ರಮವನ್ನು ಆಯೋಜಿಸಿ ನಮಗೆ ಪ್ರತಿನಿತ್ಯವೂ ಸಲಹೆ ಸೂಚನೆಗಳನ್ನು ನೀಡುತ್ತಾ ಬಂದಿರುವ ನಮ್ಮ ಅಕಾಡೆಮಿಯ ಮುಖ್ಯ ಕಾರ್ಯನಿರ್ವಹಣಾಧಿಕಾರಿಗಳಾದಂತಹ ಡಾಕ್ಟರ್ ರಮೇಶ್ ಸರ್ಗೂ ಕೂಡ ಧನ್ಯವಾದಗಳು ಸರ್. ಹಾಗೂ ಇಂದು ಹಾಜರಿದ್ದ ನನ್ನ ಎಲ್ಲ ವಿದ್ಯಾರ್ಥಿಗಳು, ನನ್ನ ಸಹೋದ್ಯೋಗಿಗಳು ಹಾಗೂ ವೆಬಿಕ್ಸ್ ವೃಂದದವರಿಗೂ ಕೂಡ ಧನ್ಯವಾದಗಳನ್ನು ತಿಳಿಸುತ್ತಾ ಒನ್ಸ್ ಅಗೇನ್ ಥ್ಯಾಂಕ್ ಯು ಅನಂಡಾಲ್. ಥ್ಯಾಂಕ್ ಯು ಮ್ಯಾಮ್.
Thank you, sir. Thank you very much, sir.
ಸೊ ನನಗೆ ಈ ಒಂದು ಟಾಪಿಕ್ ಮೇಲೆ ಮೇಡಮ್ ನನಗೆ ನೋಟ್ಸ್ ಒಂದು 4 to 5 pages ನೋಟ್ಸ್ ನನಗೆ ದಯವಿಟ್ಟು ಮೇಲ್ ಆರ್
ಕಳಿಸ್ತೀನಿ ಸರ್ ನಾನು.
ವರ್ಡ್ ಅಲ್ಲಿ ಕಳಿಸ್ಕೊಡಿ ಮೇಡಮ್ ನನಗೆ.
ಸರಿ ಸರ್, ವರ್ಡ್ ಡಾಕ್ಯುಮೆಂಟ್ ಮಾಡಿ ಕಳಿಸ್ತೀನಿ ಸರ್.
ಓಕೆ, ಥ್ಯಾಂಕ್ ಯು ಮೇಡಮ್. ಥ್ಯಾಂಕ್ ಯು ವೆರಿ ಮಚ್.
Thank you, sir. Thank you very much, sir. Thank you.